Hi, Alex. Thank you for
Hi.
joining. Hey, okay, I wasn't sure whether I did something wrong because it's like 10 and... No, okay. Thank you. That means that the attendee function is working. So I'll just wait for people to come.
Right, but is the session now?
Yeah.
Okay. Because it didn't show up in the agenda as 10. I mean, it's 10 for me.
Also, this is currently like live streaming. I don't know whether I can interrupt that. Wait. One moment. Wait. Okay, Alex, I've messaged you in the chat, just so that I'm not talking on the live stream all the time.
My name's Ellie. I'm the MC for today. So I'm going to be like telling everyone who you are.
I'm just the back-end host, so I'm just like letting people in and blah blah blah. So like,
Cool, yeah. Thanks a lot. It's cool
No.
that so many of you do this.
This is the first time for both me and is it Ariana?
Ariana, exactly.
Yeah.
Yeah.
So
I've tried to practice as much as I can so that you're not my guinea pig. Like, so hopefully, you know, it should go smoothly.
yeah, I'll definitely need some help pronouncing some of the titles as well. Hello.
Hi guys.
Hi, I'm the MC today, so it's really important that I know how to pronounce your name.
Hi MC. So my name is pronounced Nikola Jajcaj.
Yeah,
Moment. Just take 60 seconds.
Shall we test the presentations before, or like whether it works?
oh, yeah, I could stop sharing and we could quickly test if you can all share your screens.
Okay, I will stop the live stream. 1 moment.
Oh dear. Hi, everyone who's here already. We'll be starting in about 3 minutes. All right, welcome everyone to this session. We're in the theme E, which is computational modeling and techniques, and we have 3 speakers today. The format today is interactive, which means speakers will have about 5 minutes to introduce their topic and give their talk, and then we have 10 minutes to discuss those. You're all really welcome to use the chat, but also the Q&A function in the Zoom webinar to ask your questions. And we're going to kind of feed them after the talks and hopefully have a good discussion. In the Q&A panel, we'll ask you if you want to unmute yourself to ask us questions, but we can also ask on your behalf, of course. So without any further ado, I'm going to introduce our first speaker, which is Sadra Sadeh. He's based on Imperial College London, and he's going to talk about the theory of neuronal perturbation linking connectivity to coding via perturbations. Sadra, the stage is yours.
Thank you, Eleanor, but I'm not capable of sharing the screen yet. Oh, now I can. Perfect, thanks. Cool. Yeah, thank you. In this work, we try to develop a theory of how neuronal perturbations can link to connectivity and coding in cortical networks. As we know, neuronal perturbations are becoming very important and pivotal in our understanding of the brain function. And recently, our perturbation techniques are becoming more complex and more specific. For instance, in this recent study by Chetty and Harvey, they managed to stimulate single neurons in vivo in the primary visual cortex of mouse and measure the impact of such perturbations on the rest of the network. They found that the main influence of neurons on each other is suppressive. Excitatory neurons mainly suppress each other. And this suppression was in fact more for neurons with similar functional properties. For example, neurons who are preferring respond to similar preferred orientations, they respond each other more as a result of single neuron perturbations. It seems to be a bit at odds with recent connectivity data in mouse visual cortex, where we find that excitatory neurons with similar functional properties are in fact connected to each other more and more strongly. To make sense of this conundrum and to come up with a model and theory, we developed a large-scale model of cortical networks in the visual cortex with realistic receptive fields and realistic connectivity profiles as measured in experiments. We stimulated such networks in response to external stimuli in the baseline state before perturbation and after perturbing single neurons in the network. Their change in the response of other neurons in the network as a result of single neuron perturbations was then normalized by the perturbation to obtain a measure of influence. When we plotted that influence as a function of signal correlation or response similarity of neurons in the network, we observed two regimes of interactions. One regime in the intermediate range, where we observed similar feature-specific suppression as reported in the experiments, but there was also another regime of amplification for highly similar neurons. And that regime was in fact also consistent with what the experimentalists found in single neuron perturbations. We could parameterize our networks in terms of different connectivity profiles between excitatory and inhibitory neurons. And by doing a massive parameter search of the parameter space, we could observe that the main ingredient of feature-specific suppression in such networks is the presence of strong and specific E2I connectivity in these networks. We could further develop a theory of how specific motifs of E2I interactions lead to the emergence of such feature-specific interactions and suppressions. And it was very important to understand how connectome is different from perturbome in these networks, especially in networks with a strong interaction of excitation and inhibition. The theory would work for small models and simple models, but also for large-scale recurrent networks of excitatory and inhibitory neurons, where you need to consider all the higher order motifs of interaction between excitatory and inhibitory neurons. I'm happy to expand on this in the discussion if there was any further question. Moreover, we observed that the same motif of connectivity was very important for sensory processing and functional properties of the networks. We presented networks with natural images and look at how networks can distinguish between natural images. And we observed that networks with a strong E2I connectivity and a specific E2I connectivity had the more capacity to decode and encode the natural images. And that was accompanied by the emergence of nonlinearities at the population level, which was important for orthogonalization of responses and population representations. Finally, the theory and model could provide further predictions to be tested in future experiments. These are not tested yet because of technological challenges attached with them. First of all, we could shed light on the temporal evolution of single neuron influence, whereby we found that feature-specific suppressions actually emerge later in time, whereas amplifications are present at the beginning. We could also see that in double cell perturbations, when you perturb two neurons at the same time and measure the influence, you see even more suppression, which can be tested further. The theory can also be expanded to more complex situations like multiple cell or pattern perturbations, which would help us in future to map the perturbome of neuronal networks in future experiments. All of this is published now, and if you want to see further details, you can refer to the published paper, but I'm happy to answer any question that you have. In summary, model and theory, we developed a model and theory to study the effect of perturbations in cortical networks and link them to connectivity and coding. And we found that a specific EI connectivity was necessary for feature-specific suppression as measured in experiments. And we could provide further predictions and further expansions of the model and theory that could pave the way to map the perturbation of neurons in future experiments. Thank you.
Thank you very much, Sadra. We already have a question from Marcel. Marcel, if you want, you can unmute yourself and ask the question to the speaker directly, or I can relay it for you as well. So the question is, how do you relate the idea of assemblies being important functional units in the brain and the findings that suppression is the dominant mode between excitatory neurons?
I think that's in the two different regimes that I described. So you have this like dominant regime for most of the neurons, where you have the main effect is suppression, as you see here. So this like consists like a bulk of the population, like 90-something%. But then you have also some very highly like connected assemblies. So you can have different regimes like suppression and amplification based on the degree of like if you wish assembliness. Like most of the neurons are suppressing each other, but then if there is a clique of neurons who are like highly connected to each other, they can amplify each other and stand out of the crowd. But by amplifying each other and also suppressing the rest of the network at the same time. But I think another key point to answer this question is that you should look at the interaction of excitation and inhibition. If you stick to the like a traditional understanding of assemblies where you have only excitation, you cannot understand the suppression. So the most important point here is that assemblies exist not only in excitatory subnetwork, but also in the interaction of excitation and inhibition, and that's very key. Because before we were not capable of measuring that excitatory inhibitory interaction. And that's very important to pinpoint the regimes of interaction.
Thank you very much.
Thank you very much. Yeah.
Oh yeah, Marcel, did you want to ask a follow-up on that? Sorry, I just figured out how to unmute you.
Yeah, so what I wanted to ask was, what do you think, which role do inhibitory assemblies play in that? So assemblies between inhibitory neurons.
I think the first point is that it is very important to have like a similarly matched, at least some level of specificity in the inhibition and their connectivity, because if you form assemblies, they can easily go like crazy. So they can like excite each other and they dominate the network, or they go to like runaway activity. So inhibition seems to be a very good way of like the controlling system naturally. And secondly, it is important to suppress the noise, because if you just amplify the signal within the excitatory neurons, they are not isolated. Like the traditional models, they put them like just binary like subgroups, but in reality, they are connected to other subgroups as well. So when they start to activate each other, they also amplify like a non-relevant subgroups, and they start to become activated, which increases the noise. In fact, that's what we saw in the simulation that I showed you in response to natural images. So if you just put like a weak E to I, like you have a strong E to E connection, but weak to E to I, that's actually worse than feed forward. And that's because you are amplifying the noise. But when you add the strong E to E plus a strong E to I, you not only amplify yourself to a certain extent, but also like suppressing the non-relevant information. So to sum, so first you need them for stability, and secondly, you need them for increasing signal to noise ratio.
Okay, thank you. Thanks.
You're welcome. Thank you for this.
Thank you, Sandra and Marcel. We have two other questions. Oh, three other questions we're getting here. I'm going to go by order of appearance. Nicola, you've had a question. Did you want to ask that in person? And I think you're able to unmute yourself.
Yeah, sure. I just wanted to ask whether you do have an intuition why the amplification kicks in earlier than the suppression. Why there is this difference?
Yeah, we analyzed this thoroughly in the paper. Amplification emerges mainly in direct connections, whereas inhibition mainly emerges via polysynaptic interactions, and that's why it is late. So it is actually a hint. I didn't have time to go into that, but you can analyze the motifs of interaction and how amplification and inhibition emerges when you project them over the motifs, and you see that mainly the direct connections are amplification because it is the typical thing that you would expect. But then you have many multiple pathways of higher order motifs that can be either excitatory or inhibitory or disinhibitory, and we can analyze it in the network. But this prediction that they should be late rising, if it is correct, talks to the polysynaptic nature of suppression in the network.
Yeah, perfect. Thank you.
Thank you very much. Sandra, feel free, by the way, later to post a link to your paper in the chat as well. The next question, that was Daniela. No, not Daniela, sorry, Ariana. Ariana, do you want to pose your question yourself?
Hi, yeah, sure. Okay, sorry for the super unknowledgeable question, but it's the first time I've even heard many of these terms. And what I didn't fully get is the perturbome. If we would want to kind of quantify something like that in an actual in a brain, would it be defined kind of on a large network level? So, for example, would we be interested in changes in activity in say a whole functional area upon changing activity of one neuron, or would it be on a small circuit level? I didn't fully grasp that concept.
Well, it is in large scale networks, but typically what we do is we perturb a large scale number of neurons as well because of our techniques. Or because if you stimulate one neuron, it is very difficult to measure the impact of that neuron. Because the network is built of billions of neurons. It's very difficult to stimulate one neuron and effect. And that the whole beauty of the experimental result that they presented was that they could assay some sort of the effect of they could get some signal out of that noise. But it is also interesting that because it is showing us something that would have been more difficult to get with more complex pattern of perturbations. Mm-hmm. Because when you have more complex pattern of perturbation, you get more interactions, which makes it more difficult to tease out the specific interaction that you get from a single cell. So if we can get that single cell, that resolution and that specificity, we can tell something about connectivity. The perturbome is not exactly like connectivity because of all these higher order interaction, especially when you have excitation and inhibition. But it can, if you have theory and computational modeling, it can bridge the gap between connectivity and function, essentially.
Because you could then extract that from the perturb. Okay, yeah, that makes a lot of sense. Thank you.
Thank you.
Awesome. Next up is Bastian.
Thanks a lot, Satra, for the talk. I really liked the work.
Thank you.
So I was interested in, do you see any distance dependency of suppression and amplification? Because I'm aware of a lot of work, especially in visual cortex, where they assume for modeling these Mexican head-shaped profiles or this sort of stuff. So do you see something like that, or do you have anything like that in the model at all?
But the Mexican head can be assumed in two spaces. One is the physical space, which is the distance dependence of connectivity. For example, there's a paper similar to this from Allen Institute where they had this actual connectivity profile, and there they could see that distance dependence. And actually, the distance dependence is also described in the original publication, in Chetty and Harvey. So there is some distance dependence. But you can also assume a Mexican in the functional space because of that specific connectivity that I told you. So neurons with similar functional properties, independent of whether they would develop some sort of connectivity to each other. And I think that's more important for us. And that's exactly the difference of a specificity that you allow for excitatory and inhibitory neurons. And if you allow for, and this is something that we didn't present here, but we quantify that, we parameterize that in terms of the specificity of excitatory and inhibitory neurons. And you can see that it is important to have both a specificity of excitation and inhibition, but also you can play with having more specificity in excitation than inhibition. and then you get some sort of sharpening. But it is important to have some degree of specificity. So this is the slide that I was referring to. So you can play with the inhibition dominance in the network, but also with the amount of specificity of inhibition in the network. And there is a regime where you get that like feature-specific suppression. If you have only broad inhibition, you don't get it. You need some degree of specificity of inhibition to get it, but it shouldn't be also too much, because if it is too much, it kills the amplification. Sorry, I didn't have time to talk about this, but this is like thoroughly discussed in the paper. If you wanted to see more details on it, you can refer to it.
Okay, we have a little bit more time for a very brief question. Matt, I have allowed you to talk. You should be able to unmute yourself. Sadra, please be fairly quick with your answer here.
Sure, sorry.
So I'm just interested in the connectivity of how the connectivity relates to the correlation and therefore the amplification or suppression. And I just wondered, would you predict, or do you know whether the highly correlated neurons that amplify share strong sensory tuning, or are they actually sort of more stimulus invariant and might convey other information about a stimulus, for example, or not care about sensory information?
Well, we know that we don't know it from experiments, because I think the reason is that they didn't massively target all the neurons in a small scale to be able to tell that. But we know that the noise correlation is more between neurons who are like amplifying each other, which might be going back to the question that maybe the direct connections are actually the amplification, directing the amplification, whereas the inhibition is emerging via polysynaptic synapses. I'm not sure if I answered the question, but.
No, this looks great. Thanks. I'm just, it's a really cool concept, and I'm excited to see how we can sort of expand it to apply to like other sort of maybe more complex connectivity rules that we know of in Cortex. And so it's really cool. Thanks.
Thank you, guys. Moving on. Thank you so much, Sadra, one more time for this really good discussion and talk as well. Next up is Nicolas Jassai. I hope I said this correctly, from the Technical University in Berlin. And his talk is titled Towards a Large-Scale Model of Sleeping Brain, Sleep Spindles in Network Mass Models. Nicolas, are you able to share your screen with us?
Yes, I hope so.
The stage is yours.
Thank you, Eleanor, a lot for the introduction. I just want to share with you a few thoughts about how to model sleep spindles in network mass models, the work I've been doing for the last year. Let me start with the motivation. The main motivation for my work is memory consolidation, where according to the two-stage model of memory consolidation, the newly encoded episodic memories are stored in hippocampus during the day, which offers rapid storage, but unfortunately also sparse representation, which means that it has very poor generalization. Then it is thought that during the offline periods, which simply means when we are asleep, these memories are transferred to the neocortex for long-term storage. And this happens via interaction between three brain areas, and that is the neocortex, hippocampus, and thalamus. Each of these brain areas during sleep offer some canonical oscillation. For the neocortex, those are the slow oscillations around 1 hertz. The thalamus is generating sleep spindles around 12-13 hertz, and the hippocampus is generating sharp wave ripples. And when all of these three oscillations are somehow hierarchically nested, and then there is a memory replay from hippocampus to neocortex, and by this there is an information transfer. And the neocortex offers slow acquisition and is parametric, and it's very efficient in generalization. My main work was the study of slow oscillation and spindle interactions. For now, we will completely omit the hippocampus and sharp wave ripples. And there is also a secondary motivation, and that is we want to be able to somehow perturb our brain while we are sleeping and strengthen the interaction between slow oscillation and spindles, which should lead to better memory consolidation, especially in elder people. And that is important that the cortical model we are using is adapted to include perturbations via transcranial current simulation. A few words about the model. We are using two different models for two different brain areas, of course. The thalamus is conductance-based mass model with two populations. The TCR stands for thalamocortical relay neurons. It is excitatory population, and TRN stands for thalamic reticular nuclei, which is inhibitory population. Since it's conductance-based model, our main variable is the firing rate, which is a simple monotonic transfer function. And then, of course, the average membrane potential is evolving according to Hodgkin-Huxley-like dynamics, where we have intrinsic currents important for thalamus, which are the rectifying current, calcium current, and potassium leak currents. For the cortex, we chose the adaptive exponential integrate and fire neurons, but their mean field approximation. So it's also a population model, and it includes the current mediating spike frequency adaptation. We can somehow evaluate our model in 3 different levels, as I like to call it. In first level, we just have the isolated thalamus and study just the isolated thalamus. In the second level, we have our cortex driving the thalamus, but there is no feedback connection. And in the third level, we have the full thalamocortical loop. I will briefly go through all of these levels because each of these levels can bring some knowledge to us. So in the first level, we have the isolated thalamus, and in my work, I'm in general, I in general study how the full dynamic landscape looks like. That means you have your model and you try all of the parameters, all of the inputs, everything, and you see how the model behaves in different settings. So the thalamus is parameterized via the conductances, since it's conductance-based model. We have 2 conductances that are important for spindle generation. And here in these plots, the color codes the spindle density. So meaning we have the isolated thalamus and how many spindles we have. And we were able to find in the noise loss case 3 different regions in which the thalamus is making spindles, and then we crank up the noise because of course the brain is noisy, at least when we model it. And only 2 of these regions somehow survived. And of course, we have a lot of purely noise-induced spindles around with some low numbers, but in general, these 2 regions really survived. And then we did the same for the isolated cortex. And the parameterization here is not via conductances because it's a different model, but we parameterize it via input currents to both excitatory and inhibitory masses. And what is important here is this upper limit cycle, LCAE, which stands for limit cycle between adaptation and excitation. That is why it's important our model actually have some spike frequency adaptation. And this limit cycle is actually the slow oscillations. If the cortex is parameterized in this regime, it produces slow oscillations around 1 or 2 hertz. Now let's go forward and let's connect these 2 modules. So the thalamus is parameterized in the noise spindles regime. The cortex is parameterized mainly in the adaptation excitation limit cycle. And what we've done here is something like a 3-dimensional landscape. And of course, it's hard to plot the 3-dimensional landscape. So for each pair of dimensions, I do 2D projections. This is how it looks like. What is important here is this plot in the last row middle column, because this is the same parameterization as in the last slide. So the inputs to excitatory and inhibitory population, and here where the limit cycle is, now the color coding is for number of spindles. This is very nice because that means that when our cortex is parameterized in a slow oscillation regime, meaning the cortex is doing slow oscillations, we connect it to the thalamus, so the cortex is driving the thalamus, then the thalamus is spindling. And as for the typical time series from this region, we could see something like this. And the plot on the right is very nice because we can see individual spindles, how they match up. So I did a spindle detection algorithm and then plotted around their peak. And we can see here spindles in white and the cortical slow oscillations in green. And what is very nice, it seems that the spindles are initiated on down to up transition in the slow oscillation and peak in the up state. And we also observe significant phase amplitude coupling, which is very nice because this is all qualitatively consistent with EEG data. I'm running out of time, so just very briefly, when we do the full model like this, we can change the connections from both ways, the feed forward from cortex to thalamus and feedback from thalamus to cortex, and we can see how the dynamic landscape of individual modes changes upon this connection, which is very important. And the final slide, this is how the typical time series looks like in the whole thalamocortical loop. And what is beautiful is that some of the thalamic spindles here in the blue are imprinted to the cortex, which can also be seen here in the time frequency representation. So the cortex is still doing slow oscillations, but we are also seeing some imprinted spindles. And very quickly to conclusions, we have successfully connected two sleeping brain modules. And we have modeled spindles and slow oscillation interactions and their cross-frequency coupling. And it seemed to be qualitatively consistent with the EEG data. And as for the next steps, we want to see whether these conclusions would carry over to the network level, which means we want to have more cortical nodes as per structural connectivity from the DTI, and then thalamus connected and see how this would look like. Some references and acknowledgements, and thanks very much for your attention.
Thank you very much, Nicolas. You did take those 5 minutes quite literally, but we still do have time for discussion. I haven't seen any questions come in so far. So I'll give people a bit of a moment to kind of read through your conclusions again. Oh, someone's lifting their hand. Douglas, I've allowed you to talk if you wanted to ask a question.
Hi, can you hear me?
Yes.
Yes.
Hi, yeah, thanks, great talk. I was just wondering, when you measure like the incidence of spindles or slow oscillations, are these just like in a sort of in a background state, or are these like connected to what you said at the beginning of the talk related to like how these oscillations are actually some kind of replay of memories?
Well, in our model is very, very simple. It's just phenomenological. So of course we do not have real memories encoded in our model in any way. I mean, the main motivation stems from the fact that if you want to do the memory consolidation, it is a very delicate process and you need a precise timing. So it is thought that the interaction of these oscillations, the slow oscillation and spindles, are not really connected or are not conveying the actual information transfer. It's more like they are setting a stage, if you will. They provide like a clock, like a temporal windows in which the hippocampus is said, okay, now the brain is very plastic in the up state, of course, because you have a lot of firing in the cortex. So now the brain is plastic, so you can start your memory replay. And the actual memory replay has nothing to do with slow oscillation or spindles. It's a very convenient to do the memory replay then. So we are not really modeling the memory consolidation by itself. We are just modeling the interactions and how they look like and whether they agree with the observed interactions in the EEG data.
All right, thanks. Yeah, it's interesting to see that you can like sort of do this coupling regardless of any memory. Yeah.
Yes, it has been also observed that even the thalamic slices, so you have thalamus completely out of your brain and the thalamic slices are doing spindles by themselves. And also the cortex is doing slow oscillations by itself. It's just when you connect them, they start to be nested and you have some hierarchical coupling between them, which is really nice actually.
Yeah. So that's how I would usually think. Like I know of some papers where they sort of decouple these two, right? And then they show that this does not alter the frequency or the incidence of slow oscillation or spindles, but it just affects the coupling, right? I guess that's where you're going.
Yeah, exactly,
Right.
exactly. And as I was talking, the secondary motivation is that if you introduce the electrical stimulation to the brain while you are sleeping, you can actually play with this strength of this interaction. And by that, you can strengthen the memory consolidation just by playing with the strength of interaction between slow oscillation and spindles.
All right. Yeah, great. Thank you.
Thank you. Were there any other questions? And yeah, by the way, do feel free to just put your hand up as well. Like your virtual hand, and then we can help you to ask your question.
I had a small question. So
yes, please go ahead. Yeah.
How easy would be to incorporate other regions in this, like hippocampus, or is it? Do you envisage that it would be easy to capture that interaction as well in the same like type of modeling framework, or do you envisage any difficulty?
Yes, that's a very good question, because ultimately that's when I hope where this is all leading to. The thing is that formally, I mean, just writing the code and integrating and simulating this, it's kind of easy. I need to say it will. I mean, it's like 1 or 2 days, yeah. But the problem is, of course, you need to do like the full investigation, what is actually happening there. And that is extremely hard because you have so many parameters. Fortunately, both of our models are very biophysically realistic, meaning that a lot of parameters are actually biophysical, so you can get the numbers for them from the literature, right? But still, you need to do a lot of investigation. So what we are thinking of doing is the next step is really to do like full cortex plus thalamus, and we need to see whether the slow oscillations would be doing waves, because in real brain the slow oscillations are actually waves traveling from anterior parts of the brain to the posterior. And the thing is, we are not really sure whether this will hold in our model well when we connect it with the thalamus, and just then there would be a nice window to try to add some good model of hippocampus. Another problem is that usually when I was checking out some hippocampal models, you either have spiking neuron models, which won't work with our population-based models, and the population-based models of hippocampus, I haven't seen not one. So this would be more problematic, because even the model of hippocampus is not readily available. So that would be really challenging.
Thank you.
Thank you. There's one more question in the Q&A. Sorry if I mispronounced your name, Aishwarya. I've unmuted you, so you'd be able to ask this question yourself if you want. So the question is, can we also look at interactions in terms of spindles and wakefulness using these models? Brief answer, please.
Yes, spindles and wakefulness, yes, that might be possible. I mean, from the parameters I put into my models, the model is parameterized such that it is sleeping brain. But of course, changing some parameters, in particular, for example, there are this neuromodulation via acetylcholine, which is thought that this is something like waking up from the sleep. So upon changing some of the parameters, you can model this. The whole model would be as if you are not sleeping but awake. The problem is when you are awake, we cannot see slow oscillations at all, because then the main activity is faster, right? It's like alpha or beta. Only if you are sleeping, it's very slow, like slow oscillations. So you can still maybe see spindles, but not slow oscillations. Therefore, it doesn't really make sense to study slow oscillation and spindles interaction in the awake brain, because there are none.
All right, thank you very much, Nicola. This leads us just on time to our next speaker, which is Bastian Eppler. Bastian is at the Frankfurt Institute of Advanced Studies, and he'll be talking about abrupt transitions of response patterns emerging from gradual changes of network connectivity. Stage is yours.
All right, thanks, Eleanor. Hi, everyone. Today, I'm going to talk about abrupt transitions of response patterns. This work was done together with my supervisor, Matthias Gaschuber, and is heavily inspired by experiments done by Dominik Aschauer and Simon Runke. So first, for a quick motivation, we see abrupt transitions in model network responses, as well as in experimental 2-photon recordings. And an example of these recordings is here in the top right, where you can see that a certain stimulus evokes a very stable response during an imaging day, and sometimes even across days. But then at the next day, here at day 5, some completely different neurons are active. So we were wondering, how are these abrupt changes linked to changes in the underlying network? And to understand this, we want to look at the fixed point dynamics of a circuit model. And where here at time point t, a stimulus can fall into a well of the energy landscape, or an attractor, and we were wondering what is happening. So is it that the attractor landscape is only shifting slightly, which can still lead to an abrupt change in a response, or is the energy landscape changing drastically, where which of course also leads to an abrupt change. So to address this, we have a model of auditory cortex. It's very simple, and we can already with this simple model reproduce findings from mouse auditory cortex in a regime that is dominated by strong inhibition and strong recurrent connectivity. The model is just, we have some random input, we have a firing rate for each neuron, and each neuron is connected to each other. Now what we do is we add some synaptic drift. In this case, it's modeled via some diffusion model, but it doesn't really matter. It can be any synaptic drift. And what we see is, here are some example stimuli and responses that the model or the responses are pretty stable until, in this case, around time point 70, when they change abruptly. We looked at a fixed point of these of stimuli, and we can also see that these seem to be pretty stable from time 1 to 69, and then again from time 71 to 95, but there's a new fixed point emerging at this time point where we also see these abrupt changes. So we were now wondering, yeah, how can we understand these transitions? And is it this slight or subtle fixed point changes? So in this case, only the unstable fixed point between two potentials is moving, but it still makes the stimulus be remapped to another attractor? Or is it something different happening, like new attractors appearing? To investigate this, we looked at a fixed point displacement. That's the distance between fixed point at a time point t to fixed points at a time point t-1. And if we have subtle changes, the fixed point displacement is, yeah, basically 0 or small values. While when you have drastic changes, we would see a spike in the fixed point displacement. And also that's what we basically see. So the fixed point displacement here for 10 different stimuli is changing very slowly. Sometimes there are spikes, but there's quite a lot of spikes appearing at this time point where we see this remodeling of the response. And this can especially be seen very nicely in the sum that is displayed in black at the bottom. We have another problem here in this plot now, because sometimes it seems like fixed points completely disappear. So we don't see fixed point displacement anymore. And we were wondering what is happening there. So we looked at a dynamical solution of the system. And we did this by running the simulation very long. And we find basically 3 different behaviors. One would be fast convergence. That means the system is already converged and we don't see any change in the response anymore. We see sometimes slow convergence, where the model is still converging even after a long time. Or sometimes we see more complex dynamics, which could be, in this case, is a 2-cycle. It could be larger cycles, or it could even be chaotic. And what we see here is that, again, these transitions seem to occur at the same time point where we see these changes in the response. So to conclude, we have a generic firing model that can reproduce key characteristics of neural responses for strong connections and strong inhibition. We see abrupt transitions of response patterns as a result from gradual changes of network connectivity. And these transitions coincide with qualitative changes of the fixed point structure and the dynamical solution of network activity. This I want to thank my supervisor Matthias and collaborators Simon and Dominic, and you for your attention. Thanks.
Thank you very much, Bastian. We do have plenty of time for questions. Just a reminder, you can use the Q&A function, you can use the chat, or you could put your virtual hand up, and then I can see that in the attendees list and allow you to switch on your mic. Any questions for Bastian? Oh, yes. Yes, Douglas, you're...
Hi, I had just one quick question. If you could go back to your previous slide. Yeah, so the conclusions one, actually, like when you say that the it can reproduce these characteristics from like this abrupt transitions, right? And in response patterns. How I wonder how it relates to synaptic plasticity in the context of learning. So like I know you mentioned here gradual changes of network connectivity. So are you thinking of like synaptic plasticity induced by learning or some other kinds of changes in the network connectivity?
It doesn't really matter for this sort of behavior, of network behavior. So we can add any synaptic drift, we can change synapses more directly. We typically always see that if you change the synapses slightly, it does that nothing is happening until at some point there is this abrupt transition. And actually we think that it could even be a mechanism that's underlying this sort of learning that we experiment or that we experience ourselves, that typically or quite often when we learn something, we learn and learn and learn. And at some point there's this transition where we understand something. And this could be an underlying mechanism for this. And it comes in a very, very simple model. So.
Yeah, so yeah, I was just curious because like there are some very like big, abrupt transitions as you call it here, in the response of some neurons after learning. So I was wondering if it could be connected, and I guess it could be. Thanks.
Thank you. Thank you, Douglas and Bastian. There's another question. Manuel, I've allowed you to unmute yourself.
Hi, can you hear me?
Yes.
Yeah. So you mentioned learning, and I'm not sure what are these networks learning, and I would like to also ask whether you try different tasks, if they are learning any tasks. Sorry if I missed that information.
Okay, so far the networks are not learning at all. We just change them randomly. I think it would be basically the same behavior if they are learning. So as long as
there is a task, if there is a goal, probably the fixed points at some point they will be stable, completely stable, because there will be some pressure to maintain them because there is a performance to maintain, right?
Yeah, probably yes. There's a really nice work on this by Susilo, where they also came up with the method to find the fixed points that we were using. It's called opening the black box.
I know that. So for instance, you could apply this same approach to that and see if when the fixed points arise and how abrupt is. That would be interesting, I guess.
Yeah, definitely.
Okay. Thank you.
Thank you.
Thank you very much. Are there any further questions? Yeah, Sadra, go for it.
Thank you, Bastian. I was wondering about the time scale of this transition. Maybe I missed it, and which parameters define that? Because, for example, in visual cortex, the time scale of this transition was a big issue for a while.
Yeah, thanks a lot. It's a very good question. Actually, the time scale doesn't really matter. We find these transitions on any time scale. So at least any we looked at. We looked at very large time scales, of course, but also at very small. And we couldn't they always seem to be abrupt, which is quite fascinating.
Thank you.
Thank you. We do have another 5 minutes before Neuromatch kicks us out of the room. So if there's any remaining questions, we do have time for them. Well, with this, thank you again to all our speakers in this session. This was definitely really interesting, something really out of my field. So I thought it was interesting to hear some new things and learn new things today. Thank you so much, and hopefully see you around in the other talks going on this week. Bye, everyone.
Bye-bye.
Thanks. Bye.
Bye.
Thank you, Eleanor.
Thanks.
Now we are live on YouTube.
Oh, great. We still have about a couple of minutes. All right, so let's get started here. Welcome everyone. My name is Shankar. Along with me, I have Firuzan Farhani. We are excited to hear about the role of prefrontal cortex in sensory processing from 3 distinguished speakers. So let me just go over the format of the talk. It's a traditional talk, which means we have about 12 minutes for the talk and about 3 minutes for question and answers. Please ask your question and answers in the Q&A using the Q&A option in Zoom, and I will read them out to the panelists. And please vote for the questions that you feel are pretty interesting as well. And this is supposed to be a very engaging session, so please feel free to chat with the panelists or attendees using the chat button. And welcome again. So first up, we have Dr. Rafiq Huda from Rutgers, and I'll just keep the introduction short so that we have a lot of time for the talk and the question and answers. So please take it away, Rafiq.
Thank you, Shankar. I'm really happy to be here, tell you a little bit about our recent work looking at the role of distinct prefrontal output pathways in sensory motor behaviors. So I'm going to start by just plugging the fact that I just started a new lab and we're hiring at all levels. So if the work I'm about to describe to you sounds interesting, please reach out and we can carry the conversation forward from there. Sensory motor behaviors are a crucial building block of voluntary behavior. And although they may seem simple, they require at least 2 distinct capabilities. Number 1 is attention, which is the ability to prioritize sensory information from the world in order to act upon it. And then number 2 is action selection, which is the ability to select a particular action that's appropriate for the given condition from a large behavioral repertoire. And the prefrontal cortex has long thought to be important for both of these processes. And here I'm just showing you a few collections of different brain areas that have been implicated in sensory motor decisions. And what I wanted to impress by giving a rough idea of the anatomical connectivity between these structures is that the PFC really is connected to all of these structures, either directly or indirectly. So again, based on this connectivity and work that stretches over the past few decades, the idea has emerged that the prefrontal cortex is a crucial anatomical and functional hub for coordinating various aspects of sensory motor decisions. But this really raises a fundamental question, which is what we hope to address with our work here. And that question is, how can a single brain structure contribute to both sensory processing and action selection underlying sensory motor behaviors? And so I'm going to start off by giving you what we think the answer is. In our work, what we have found is that in the anterior cingulate cortex subdivision of the prefrontal cortex, which is the area we studied in the mouse, there are two non-overlapping population of projection neurons. One population of projection neuron that targets the midbrain motor structure of the superior colliculus, and another population that targets the visual cortex. And based on the evidence that I'll briefly go over today, what we would like to suggest is that the projections to the superior colliculus may be fulfilling this role in action selection, whereas the projections to the visual cortex may play a role in sensory processing. Okay, so we started this work by just doing some anatomy, because if the hypothesis is that these two functions may be subserved by two distinct anatomical pathways, then you need to make sure that's actually the case. So we turned to dual color rabies viral tracing, in which we injected a red fluorophore expressing rabies virus in the superior colliculus and a green rabies virus a rabies virus expressing GFP in the visual cortex, and then we just did simple histology to see where are the neurons that projected these two downstream areas. And what we found is that in the ACC, there are two completely non-overlapping population of projection neurons. So in red labeled are ACC neurons that projected the superior colliculus, in green the cells that project to the visual cortex. So then this provides kind of an anatomical substrate for assessing whether functional differentiation is subserved by these two distinct pathways. Okay, so in order to actually look at this, we developed a visually guided action selection task. So in this task, head-fixed mice are head fixed in front of a screen on which they're shown a visual cursor that appears either on the right or the left side of the screen, and the mice are supposed to report their detection of these visual cues by moving it to the center of the screen by rotating a trackball. So this task assesses sensory processing because you have these lateralized visual cues that have to be detected, and it assesses action selection because the animals are supposed to select one of the two actions correctly in order to receive a drop of water as a reward. Okay, so the sensory selection, the sensory processing aspects is quite clear. The mice are shown these lateralized visual cues, but of course these ball rotations that the animals are making are rather artificial, right? It's not like naive mice are sitting somewhere in the wild rotating balls left and right. And so we were wondering how this might relate to more naturalistic behavior. So what we did is we turned to deep lab cut. We recorded mice while they were rotating the ball while head fixed, and the same mice when they were locomoting while they were freely moving. And we compared their forepaw positions in these two situations using deep lab cut. Okay, so what we found is that when freely moving mice, when they're turning, let's say, for example, leftward, the way they turn is by positioning their paws opposite to the side of the turn. So when a mouse is turning leftward, on average, their left and right paw position is going to be to the right, and vice versa for rightward turns. And similarly, you can imagine that when the mice are rotating the ball, of course, they're head fixed, so their body axis is not moving, but they're rotating the ball counterclockwise by moving their forepaw in one direction or the other. So based on this kind of analysis, what we would like to suggest is that rightward turns in the freely moving condition, based on how the forepaws of the mice are moving, are actually akin to counterclockwise rotations of the trackball. And this will become important as I go through some of the experiments that we have done. Okay, so kind of as a brief summary of what the task is, the animals are shown one of two cues. A visual cue can appear on the left or the right side of the screen, and the correct response for these cues is a contraversive action or an ipsiversive action. Again, these actions are defined relative to the left hemisphere. And so essentially, in this case, a contraversive action is similar to a freely moving animal moving rightward. Okay, so of course, we found these projection neurons in the ACC that target the superior colliculus. We know from a large body of work that superior colliculus is quite important for orienting behavior. So the first thing that we did is we wanted to see if the superior colliculus is involved in this task. As you would expect from the forepaw tracking analysis that we did, it turns out that when we inactivate the sphere colliculus to inactivate the sphere colliculus, what we did here is we injected a virus in the sphere colliculus that expresses JAWS, and then on a subset of trials, we inactivated by shining orange light. So then we can compare what is the effect of SC inactivation on non-laser trial and laser trials. And essentially what we found is that when we inactivate the sphere colliculus, there's a huge deficit whenever a contraversive action is the correct response to be made. And remarkably, this was not only during a visually guided action selection task. If we look at mice when they're just untrained mice that are just spontaneously moving the ball without being trained to associate any cue with those movements, we again found that inactivating the sphere colliculus decreases contraversive action. So together, this shows that the SC is quite important for facilitating contraversive actions in this task, as you would expect from freely moving literature. Okay, so what information might the ACC be sending to the sphere colliculus? So to address this issue, we turned to 2-photon calcium imaging, in which we could selectively measure the responses of these projection neurons. And I'm just going to summarize the results that we found here. Surprisingly, what we found is that the ACC outputs to the sphere colliculus actually relays information about ipsiversive actions, not contraversive actions, as you might expect from the SC inactivation results. And so to look at the functional relevance of this information that we recorded physiologically, we turned to projection-specific inactivation. We expressed a virus expressing JAWS in the ACC, and then implanted an optic fiber cannula over the sphere colliculus to selectively inactivate only this projection pathway. And in contrast to the inactivation of the SC, what we found is that the ACC SC inactivation actually leads to a decrease in ipsiversive actions. And so exactly why these two effects are opposite to each other, we're still exploring. And again, just to plug my lab, we'll be looking at in more detail. But coming back to the issue at hand, what is the role of the ACC-SC and the ACC-VC pathway in sensory and motor processing? And so again, we turn to projection-specific inactivation, targeting the ACC-VC pathway this time. And what we found is that inactivating this pathway, instead of having an effect on the ipsiversive action as we did with the ACC-SC pathway, now we see a deficit on performance whenever the animal is shown a right visual cue. So again, if the animal is performing the task on a right visual cue, we inactivate the left ACC-VC pathway and we get a behavioral deficit. But these experiments on their own don't really help us distinguish whether these pathways are playing a role in the sensory part of the behavior or the motor part of the behavior. So what we reasoned is, instead, if we train mice on a reverse sensory motor contingency, so that now the visual cue that previously cued an ipsi action now instead cues a contra action. And then if we look at the effects of inactivating these two pathways, then we might be able to better discern whether one pathway plays a sensory role or a motor role. The hypothesis here would be that if a specific pathway plays a sensory role, then the effect of inactivation in the two tasks should be exactly the same. Whereas if a particular pathway plays a motor role, then inactivating this pathway in the reverse sensory motor contingency should switch the deficit. And that's indeed what we find for the ACC-SC pathway. So if you remember, when we inactivated the ACC-SC pathway in the previous task, what we found is a deficit on left cue trials. But now instead, we find a deficit on right cue trials. But in both tasks, what we find consistently is that whenever the correct response is an ipsiversive action, there is a deficit with ACC-SC inactivation. With the ACC-VC inactivation, we find that regardless of which task the animal is doing, there's always a deficit on right cue trials. Again, suggesting that this pathway plays more of a sensory role. So we can kind of look at this all together by combining.
Rafiq, you have 1 minute left.
Perfect, thanks. So we can look at we can combine data across both tasks and then see how inactivating each of these two pathways affects behavioral performance whenever a right stimulus is cueing actions versus behavioral performance whenever a contra action has to be made. And what we find is that inactivating the ACC-VC pathway consistently leads across both tasks to a deficit whenever the animal has to respond to a right stimulus, whereas inactivating the ACC-SC pathway leads to a deficit whenever the animal has to make a specific action. So again, to summarize everything I've told you, we think based on the results that I presented today that there are two non-overlapping population projection neurons in the ACC. One population that targets the visual cortex, another that targets the superior colliculus, and the role of these projection pathways seem to be in sensory processing and action selection. Of course, this might be advantageous because this allows the prefrontal cortex to independently control two complementary roles that underlie sensory motor behaviors. And I want to just end by thanking all the people that contributed to the work, including my postdoc advisor, Mrganka Sir, and many people in Mrganka's lab who helped along with this project. And I again want to reiterate that we're a new lab. We'll be working on this and many other questions, and come join us. I'm happy to take any questions.
So that's the applause you hear on behalf of the attendees. All right. So we have a couple of questions. So the first one is from Eyal Kimchi, who says it's always great to have a chance to hear about your work. And is there any layer distinction between ACC and SC projecting ACC neurons?
So the SC projecting ACC neurons reside solely in layer 5. The VC projecting ACC neurons can be in layer 5 as well as in layer 2-3. The imaging that we have done, which actually didn't really even have a chance to show much of, was restricted to layer 5.
Got it. And next we have a question from Lawrence Hunt. Is there any way to assess the local connectivity of the SC projecting and VC projecting neurons to identify whether they form local subnetworks within the ACC?
Yeah, that's a fascinating question, and it is a direction that that we'll actually be taking. So one strategy, well, of course you could do it in slice, but if you want to do it in a behaving animal, one strategy would be to do disynaptic tracing and then imaging locally, right? So for example, if you were to put a retrograde virus in the superior colliculus and then express your monosynaptic rabies, AAV vectors in the ACC itself, then that would label locally all the neurons that projected the ACC SC cells or the ACC VC cells, and then we can label them that way and look at what they're doing during the behavior. One caveat with this is that at least the previous generation of rabies viral vectors are toxic, but there are people are devising strategies for non-toxic labeling of these, and so this would be this would be it's a great question, and it is doable. We just haven't done it yet.
Great. Okay. Thank you again. Thank you for the talk.
Thanks.
So next up we have Dr. Abhishek Banerjee from Newcastle University. Abhishek, please take it away.
Okay, I'm unmuted. Can you hear me all? Great. All right. So thank you very much for the introduction. I would just like to quickly sort of move on. It's nice that Rafiq presented his talk right before me, because he mostly talked about sensory motor decision making that are based on sensory motor transformation. So here I'm going to talk about a slightly different thing, which involves sort of certain cognitive switches. And of course, the theme of the talk would be quite similar, which is the theme of the symposium, which is the prefrontal control and gating of flexible sort of sensory processing. And I've also started my own lab recently in the Newcastle University in the UK. So when we talk about sort of value-guided decision making, we generally talk about inferring meaning of not the sensory stimulus itself only, but also what this sensory stimulus outcome is. So perceptual decision making, as we know, or goal-directed learning, it involves multiple cognitive maps, and these maps are both cortico-cortical, cortico-subcortical maps. They are constantly talking to each other in a distributed brain region. But animals do not only make decisions. They evaluate and switch between decisions, between appropriate sort of goal-directed strategies, and they do so by a method, sort of a way of depicting this would be sort of a stimulus action outcome evaluation. So it's sort of less clear how prefrontal neuronal populations or subpopulations actually encode decision variables during a flexible decision-making task, and how their responses flexibly remaps from one sensory outcome modalities to another ones during value-guided decisions. And also another very interesting question is, we know some of these prefrontal regions, including SEC that Rafiq talked about, other areas such as orbitofrontal cortex, medial prefrontal cortex, they sort of encode decision variables, but how they specifically control primary sensory areas in a sort of a hierarchical, guided, reinforcement-driven manner to support value-guided behavior is sort of unclear. So what we did in order to address this question is we designed a task that's based on sort of reversal learning of a stimulus outcome association. So reversal learning involves adaptation of behavior according to changes in stimulus outcome contingencies. So what you see here on the left is as a mouse, again, a kind of a 2 months old, 2 to 2.5 months old mice that are, again, head fixed. And we are presenting them with distinct tactile cues. So different types of sandpaper textures that you see here. Like this is a coarse sandpaper. This is kind of a fine sandpaper that they have to distinguish in order to, they are water scheduled animals in order to win water reward. They're head fixed, and we are measuring other behavioral variables. But as you can see, since it's a go-no-go task of a tactile discrimination-based go-no-go task, the trial structure is somewhat like this. You have a baseline time when the animal has to sort of wait, and then there is a stimulus presentation. In this case, a tactile sort of cue comes towards the animal. The animal has to make a choice between a go and a no-go stimuli, and lick or withheld licking in order to implement, in order to win a reward or to avoid a punishment. So as you can see, during a go-no-go task, you have 4 different outcomes. If you have a target texture that leads to a winning of a reward, which is a hit, and they can miss the target texture, which is a miss, followed by a false alarm when they incorrectly make a decision, and correct rejection when they correctly avoid the non-target texture. So if the animals, sort of we train the animals individually on this task, and this is a single mouse that I'm showing here, if you can see that the animals actually learn the task quite nicely. Their hit and correct rejection rate over the days of training actually increases and almost reaches the 100% level. And their false alarm rate or their errors actually go down during learning. So in order to understand or study flexible decision making, what we do now is, once the animal learns the task perfectly well, we switch the rule, which means if the texture 1 was giving reward initially, they learned this outcome, stimulus outcome contingency. After they have learned that particular stimulus does not give reward anymore, it becomes a no-go. But texture 2, a no-go texture, now gives them reward. So, and as you can see, a single mouse actually relearned the task fairly well. If you see a bunch of animals, they can also relearn the task from learning through reversal. And each of these phases undergoes 2 different phases themselves. One is learning naive through expert, and reversal naive through expert. So animals actually learn this sensory discrimination task, a cue-guided task, and they adaptively change their behavior. So this is a pure sensory task. And what we were interested in is to really look at some of these prefrontal areas that play an important role in cognitive processing of information or stimulus outcome associations, invoking rule-based strategies, if they play an important role in these type of tasks. And I can cut short and right away tell you that selective silencing of a very particular brain structure in the prefrontal area, which is the lateral part of orbitofrontal cortex, impairs reversal learning. It has been seen in other modalities as well, in other animals as well. It's a very particular structure in the brain that plays an important role in stimulus outcome association. So OFC is a key higher order association cortex that has both projections to subcortical structures as well as sensory cortices to implement these type of learned association related rules. So what we did is, so as you can see from Marie Carlin's review, the OFC or the orbital areas are located quite deep down in the frontal part of the cortex. in order to understand what is happening in the brain, we prepared a novel preparation through which we started imaging these neurons using 2-photon imaging through a green lens that was sort of inserted through a metallic cannula and a cover glass that's at the base. So we are imaging lateral orbital cortical neurons throughout behavior. So how do these neurons respond? So this is an example neuron. You can see that I have aligned their trial-related calcium responses according to the learning and reversal. So I asked you sort of to remember that the learning naive through learning expert is the behavioral sort of output, and reversal also has a similar reversal naive through reversal expert. So I guess what you can appreciate from here is one very interesting thing happens as soon as we switch the rule, which is this white line that's here. So that most of these OFC neurons, that you can see the population average responses here, a lot of these OFC neurons, including the example ones here, starts responding to the newly rewarded context. So whenever there is a mismatch of a stimulus outcome evaluation, through evaluation, stimulus outcome contingency, these neurons starts encoding that sort of a value mismatch or value prediction error responses. It's not seen for the correct rejection responses. It's very specific to the hit responses that are leading to a reward. Now, since we are doing 2-photon imaging of individual of these neurons in OFC, as well as in the primary sensory cortex, of course, that you can see the sensory neurons in comparison with this predictive responses in reversal naive phase for OFC, it shows actually an opposite pattern of responses. Sensory neurons in a very similar way, it's as primary sensory neurons, shows a learning induced responses. But then as soon as there is a rule switch, there is not much happening in the sensory, primary somatosensory cortical neurons. But they completely remap their responses at a later stage during training when the D prime or the behavioral scoring has gone up again. The animals have relearned or switched the behavior. So this is extremely interesting that in a same task dependent manner, there are two areas that are completely talking in a very different way. Now, since we record these neurons longitudinally through a cortical window preparation and a green lens preparation, you can really study all of these individual neurons, hundreds of these neurons individually. And I want to highlight two important things here. One is the response alteration. So you can really study how during distinct behavioral phases, all these different neurons are being engaged or disengaged, right? So I want to highlight two things here. These two cell classes of the populations. These are outcome selective neurons and gain selectivity neurons. So how do we measure this? So what we did here is we plotted the responses of these neurons before and after of a reversal. So as you can see, the most important bit for OFC neuron was this transition between learning expert to reversal naive, when cells are mostly responsive. Whereas for S1, the transition between learning expert to reversal expert is the most important phase because the remapping has completely happened at that phase. So here what we see is these outcome selective neurons that are responding in both cases before and after reversal. So they are the ones that are there understanding this value mismatch representation, and they can actually encode or guide the rest of the brain perhaps to understand that there has been a different stimulus leading to a different outcome, and they are understanding this encoding. So if we now look at plot these comparison between learning expert reversal naive through learning expert and reversal expert, if we plot these neurons individually, and I would like to highlight the blue and the red neurons that you see here. When we look at the somatosensory cortical neurons, these are S1 neurons, primary sensory neurons. When they are if you look at their responses, what is extremely striking is during this transition from learning expert to reversal naive, immediately after reversal, when the S1 neurons are not so much, they're not so sure about how to respond, their responses are—
Abhishek, you have 1 minute left.
Yeah, great. So they're all sort of around these 0 points in an ROC plot. But all of these same neurons actually completely remap when the animals relearn the task and goes to the next level when they become reversal expert. Now, our guiding hypothesis is there is this massive remapping of reconfiguring of responses in primary somatosensory cortical areas. So our hypothesis was if we silence orbitofrontal cortex, silence or remove these value prediction error responses from the lateral OFC, whether a similar kind of responses arise in the primary somatosensory cortex. So we silence OFC, a lateral part of OFC, using DREAD receptors, and we silenced OFC after reversal or during rule switch, right after reversal. And I guess what you can appreciate here is to show you this marginal distributions of these cells. So this is the reversal learning expert to reversal expert marginal distribution. I'm plotting it in a slightly different way here. So this is S1 neuron. The pink one is reversal naive through, red one is reversal expert. There is a massive rightward shift from the previous contingency -1 to new contingency +1. This is in primary somatosensory cortical neurons without OFC silencing. If we do now the same experiment and silence OFC and record from the same S1 neurons, you see there is actually a complete overlap of these two. There is no rightward shift, no shift towards the primary somatosensory cortical neurons towards the new contingency. So this basically sums up really what I had to say, which is really during sensory processing, the sensory input, sensory learning dependent responses in primary somatosensory cortex builds up responses through this feed-forward mechanism in the lateral OFC, which is a key area for value-guided decision-making. And it computes, subpopulation of neuron computes this value prediction error responses. And through this teaching signal, feedback teaching signal that's coming from OFC to S1, together with many other areas, I should point out, that actually implement some of this responses. Just to sum up, again, OFC is an important
Abhishek,
hub.
we are running out of time quickly, so can you just
This
quickly
is the
wrap
last
it up?
slide, so
Okay.
I'm going to end. So OFC is a key hub for value-guided decisions. Silencing OFC impairs reversal learning, and neurons in the OFC actually plays a very important role providing value prediction error responses. And a functional subpopulation of these neurons that are outcome selective neurons that I showed in OFC and S1 signal reward expectancies by integrating the reward history. So I'll stop here. You can see more of this on this recent paper that we published. And I would have to acknowledge Fridjof, whose lab this work was done in Zurich, and my students here in Newcastle and also in Zurich. Thank you.
Thanks, Abhishek. That was a very exciting talk. So we have some questions. Oops. The first one is from Lawrence. Do your neurons in OFC have mixed selectivity for the different features of the task? Or are there distinct clusters of neurons which are selective for individual features?
That's a very good question. We do have very distinct types of neurons. There are some neurons that actually encode for sensory features as well. But a major fraction of these neurons are actually outcome selective neurons, which is kind of sort of expected. But we are showing it for the almost, I guess, for the first time that individual neurons are encoding these type of features.
Perfect. So the next one is from Mehmet. Great talk. What happens if you inactivate ventral OFC in this task?
We have silenced the medial part of OFC. We have, and it does not have any effect on this particular form of reversal task, but it plays an important role in other forms of reward-driven learning.
All right, great. Thanks again for the wonderful talk. Applause on behalf of the attendees. And next up, we have Philip Cohen from UCL. Please take it away, Philip.
Great, thanks very much. Everyone can hear me okay? Brilliant. Okay, so I'm going to be talking about the role of frontal cortex in multisensory decision-making. Thanks very much for tuning in to listen. This is work I've been doing over the course of my postdoc with Matteo Carandini and Kenneth Harris. So the thing I'm particularly interested in throughout my postdoc, apart from PowerPoint slide transitions, is audio-visual integration. So that's a type of integration that all of us use all of the time and is particularly useful in noisy environments. So some of you who are old enough may remember when we used to physically gather in these kind of noisy environments to look at scientific presentations. And I'm interested in asking which parts of the brain are involved in taking auditory and visual signals from a poster presenter, for example, and combining that information in order to understand what's going on in the natural world. And I want to use the mouse model system to do this. So I'm going to hopefully answer 3 questions for you today. The first is, how do mice actually integrate visual and auditory information? Then we're going to look at which regions of dorsal cortex are actually required for this integration. And finally, we're going to ask how neurons in those regions actually represent audiovisual stimuli. So in order to do any of those things, I need an audiovisual task, and the one I developed for mice works as follows. Mice are sitting in this behavioral chamber with their forepaws resting on a wheel that they can turn to the left or to the right. We can present visual stimuli using these screens and auditory stimuli using this array of speakers. Now, mice have to hold the wheel still to initiate a trial. And when they do that, a stimulus will appear on the left or the right. And it can be a visual stimulus, an auditory stimulus, or both presented together. And in every case, the job of the mouse is the same. It needs to turn that wheel to move those stimuli to the center, and then it's going to get a water reward. If it does this correctly on a coherent trial, but we can also have trials which are incoherent, where the visual and auditory stimuli are presented on opposite sides. And in this scenario, the mouse doesn't know what to do and we reward randomly. So there is no correct response, but the mouse should always make a response. So what does the behavior actually look like? I'll start by showing you this in this kind of psychometric framework, where we have the fraction of rightward choices on the y-axis and the contrast going from left to right contrast on the x-axis. And we'll look at the behavior from one mouse. And to begin with, let's look at trials where the auditory stimulus was in the center. So it provides no left-right information, and we think of these as visual-only trials. And in that case, you can see that as the contrast goes from left to right, the fraction of rightward choices this mouse makes increases. Now, we know how to model these kind of visual behaviors in our lab pretty well. We can use a simple model where the log odds of the mouse choosing right are based on a bias term and a vision term. So there's only one stimulus variable here, the visual contrast, and 3 parameters, the bias, the visual scaling, and the contrast saturation. And when we fit this model to this mouse, it does a very good job. So how can we incorporate our extra auditory condition into this kind of framework? So let's start with these 2 conditions where the visual contrast is 0, and we have auditory on the right in red or auditory on the left in blue. And the simplest way we can imagine kind of incorporating this into this model is to just add an audition term, where we've added one fit parameter, the scaling, and one stimulus variable, the auditory azimuth. And to see what this does to our model, it's actually easier to switch frameworks slightly and look at the log odds of choosing right on the y-axis versus the contrast raised to the saturation parameter on the x-axis. And when we do this, you can see our curve becomes a straight line. And so hopefully it's intuitive that by adding these auditory, this auditory term, we're effectively adding 2 new lines to this plot. And now the question is, if mice are actually using this additive strategy to combine visual and auditory information to solve this task, then we would expect all the remaining points to lie on these 2 lines, all the coherent and conflicting combinations of auditory and visual stimuli. And in fact, that's exactly what we see. And I'll switch back to the psychometric curve for those who are more familiar with that. So this is really nice, but it's just one mouse. What about other mice? Are they all solving this task with this additive strategy? And to illustrate that for you, I'm going to show you the model fit for 20 different mice. And we'll start with the mice that are most visual and end with the mice that are most auditory. And what you can see here is in all these different mice, this model is really doing an extremely good job of fitting the behavior. And we can actually capture 97% of the variance in the average behavior of these mice. So hopefully that's shown you that an additive model is fully capturing this behavior, and mice are using this additive strategy in order to solve this audiovisual task. So now we can move on to our second question, which is which regions of dorsal cortex are actually required for this integration. And to look at that, we used an optogenetic strategy, first shown in this Svoboda lab, where we're going to inactivate from the moment of stimulus onset until the end of the trial. And we're going to do this in a grid across the entire of dorsal cortex while mice perform the task. And then we can ask whether all these different areas of dorsal cortex that we're kind of interested in, whether inactivating any of these regions is going to have an effect on the task performance. So to look at that, I'm going to start by showing you just visual trials. And what I'm showing you here is for each point at which we inactivated, what is the change in the fraction of rightward choices the mouse made during inactivation. And you can see if it's red, that's an increase in the fraction of rightward choices, and blue is an increase in the fraction of leftward choices. And the size of the dot indicates the significance. So hopefully you can see that when the visual stimulus is on the left, we see an increase in the fraction of rightward choices when we inactivate contralateral visual cortex or contralateral frontal cortex. And the reverse is true when the visual stimulus is on the right. So this was really nice because it replicates previous work in our lab using a visual-only task by Zatkahass and Steinmetz, and so indicates that everything is working well with this inactivation strategy. And now we can have the confidence to go on and say, all right, what happens on auditory and multisensory trials? So in auditory trials, we saw something rather pleasingly analogous, which is that when the auditory stimulus is presented on the left, we saw that we increased the fraction of rightward choices when we inactivated either the contralateral frontal cortex or contralateral auditory regions. And the reverse was true again when the auditory stimulus was on the right. So what's going to happen now if we look at combinations of these two different modalities? Are we going to see new regions of the brain being recruited to deal with audiovisual trials? And in fact, that's not what we saw. So if we look at these conflict trials, what I hope you can see is that the effects of inactivation when both stimuli are present is effectively the sum of the effects of the respective unisensory trials. And so we don't really see any new regions of the brain like parietal cortex or retrosplenial cortex being recruited to deal with these audiovisual conflicts. So from this inactivation study, we concluded that auditory and visual cortex were required for auditory and visual trials respectively, but the only part of the brain that seemed to be required in every single trial type was this region of frontal cortex called MOS. And so this seemed like a natural place for us to record neural activity and ask how neurons in that region actually represent auditory and visual stimuli. So in order to do that, we used NeuroPixels probes, and we recorded several penetrations across several different mice. And in total, we gathered data from 2,000 cells in MOS and about 10,000 cells in other brain regions while mice were performing this audiovisual task. And so to give you an idea of what the data look like, I'm going to first show you 3 example neurons from MOS itself. And so we'll start with an example auditory neuron, and I'll show you trials where the auditory stimulus was presented on the right. So what I'm going to show you in this raster is trials where the mouse made a rightward choice in red, and when it made a leftward choice in blue. The stimulus onset is the dashed line, and the black points indicate the movement onset. Hopefully you can see from this neuron that it responds robustly after the stimulus, doesn't care about the movement, and doesn't really care about the direction. And that's pretty clear as well from the PSTH. Now I can show you adding a visual left stimulus or a visual right stimulus here, and you can see it doesn't really make a difference. There's still a robust response, and it still doesn't care about the choice of the mouse. We can see visual neurons that look very similar. Here I'm keeping the visual stimulus on the left and moving the auditory stimulus around. And again, we see that a robust response after the onset, but it doesn't care about the choice of the animal. But we do see other neurons that care about choice. So here's an example of what we call a choice neuron. Again, I'm going to show you auditory on the right, but now hopefully you can see that there's a bigger response when the mouse makes a rightward choice than when it makes a leftward choice. And that's even clearer in the PSTH. And if we look at some other stimulus conditions, again, we can see that this neuron doesn't really care about the change in the stimulus. It's always responding more for rightward choices. So this is really nice. We have this region of the brain that is involved in the task, and it seems to have neurons that reflect information it would need to solve the task. But is this true of any region of frontal cortex, or is there something special about MOS? And to ask that, we used a very simple decoding strategy where we took a 100 millisecond window before each movement onset and asked, based on the neural activity in that window, how well can we predict whether the mouse is going to go left or right? And we did this for various different brain regions. I'm showing you the accuracy relative to baseline, and the small dots are individual sessions. And we accounted for the number of neurons in each brain region here. And what we observed was that MOS was indeed the strongest predictor of choice. And that's compared to other regions like anterior cingulate, prelimbic, and all the other regions we recorded while mice performed the task. So this really does indicate there's something special about MOS. And then finally, we wanted to ask, okay, we have this additive strategy that mice are using in the behavior. What if we take the neurons that are sensitive to auditory and visual stimuli in MOS and train those neurons to solve the task? Will we recreate this additive behavior, and will we recreate the behavioral things we observed from the mice? Unfortunately, I don't have time to go through the model in detail for you here, so I'm just going to give you a glimpse of it, and I'm happy to talk more afterwards. So we trained a model to perform this task using the neurons in MOS. And we can see that this model successfully separates out the different trial types. And if we look at the log odds of this model choosing right or left, when we give it different trial types, we can see that the log odds look very similar to the kind of things we observe in the mouse behavior. And that's, it also recreates other behavioral results that unfortunately I don't have time to tell you about. So with that, I'll just conclude by saying that I've shown you that an additive model fully captures the behavior of mice on this audio-visual task, and they seem to be using an additive strategy to solve the task. Auditory and visual cortices seem to be involved in their respective modalities, but only this frontal region MOS seems to be involved in all trials. And MOS seems to have the strongest representation of upcoming choice compared with all the other regions we recorded. And a drift diffusion model based on the neurons in that region of the brain replicates numerous behavioral results. And with that, I'd just like to thank everyone for listening, thank my co-authors and Matteo and Kenneth again for their support in this project. And I'm happy to answer any further questions.
Thanks for the wonderful talk, Philip. We have some questions. The first one is from Martin. How do you distinguish these neuron types statistically? Multiple regression sliding window analysis, is that one?
Yes, so basically the way we distinguish the neurons at the moment is by using a window after stimulus onset. And we basically use a shuffle analysis. So we're comparing the response of the neuron in the different conditions when it's unshuffled, and we compare that to a shuffled distribution where we shuffle the trial condition and the choice that the animal makes. And then we see if this neuron significantly predicts, for example, whether the auditory stimulus was on the left versus right compared with that shuffle. But yeah, I'm sorry, that wasn't a particularly clear explanation, but again, I'm happy to chat afterwards if it didn't answer the question.
Got it. Next, we have a question from Shresh. How about incorporating olfactory sensory trials in the paradigm as well?
Yeah, it's a cool idea. It's difficult enough so far to get a robust audio-visual behavior, but it is really interesting question how other sensory stimuli would combine. And yeah, we certainly haven't done it yet, but it's an interesting idea.
Cool. The next one we have from Sevji. How do you predict the choice of the animal decoding algorithm?
Right. So how do we predict the choice in basically this slide? So essentially, yeah, I still don't think I have time to go through it in all the detail. But what we do here is basically take neurons that are sensitive to auditory left-right or visual left-right, and we select those neurons, and then we basically train our model in order on different trial types, for example, all the different trial types that you can see on the left. And then based on cross-validation, we basically feed in trial types, and we ask what is the fraction of times when we feed in those particular stimuli that this model chooses the leftward choice versus the rightward choice. So again, unfortunately, I'm sorry I don't have time to go into all the nitty gritty, but that's kind of a summary of how we get those model results you can see on the slide there.
Right. So we had two more questions, but we might get kicked out of the session. So we'll try to see if we can answer that within the time. So this one's from Sandra. It says, great talk. What are roughly the proportion of sensory versus choice responsive neurons you observe in MOS?
Yeah, so very roughly, we see around 5% of MOS neurons that are sensitive to choice. And if we were to summarize as all sensory neurons, like visual, auditory, or responding to both, then it's probably around the same number, 4 to 5%. So kind of in that kind of single digit region. Yeah.
The last one is from Gonzalo. In OMC, do you see more neurons dedicated to the visual modality or the auditory modality?
So it's a good question. There's a slight subtlety, which is that if the question was purely does the neuron detect auditory stimuli or visual stimuli, then we see more neurons that detect the presence of an auditory stimuli. But if we then look at whether a neuron can detect whether a stimulus was on the left versus whether the stimulus was on the right, then we see roughly equal numbers of auditory and visual stimuli. So there's a slight subtlety, but in terms of like solving the task and the neurons that go into this model, it's roughly the same number of each. Yeah.
So that's all. Let's thank again our panelists for this wonderful session. And please feel free to follow them on Twitter. And if that's it, and thanks again for the panelists. And that's about it. Thank you.
Bye-bye.
Thank you. Bye.
Bye. Thanks for hosting.
Sure. See you.
Let me know when it's done.
All right. Yeah, I think it's up.
Perfect. Well, hi, everyone. I'm Richard Nogue from the University of Ottawa. Today, we have 3 exciting talks on connectivity. These are interactive talks, meaning you have 5 minutes to come up with the questions you'll ask in the 10 minutes that are following. First up, we have Daniel Titherich from the University of Bristol, who is going to talk about the activation of 5-HT2A receptors and their effect on the cortical limbic functional connectivity. Take it up.
Hi, my name is Dan Titherich. I'm a psychiatrist by training, but I'm currently doing a PhD funded by the MRC. And today I'm going to be talking about some data that was collected by a postdoc in the lab I'm working in, looking at a 5-HT2A agonist called 25I. Why are 5-HT2A receptor drugs interesting? Well, they're the target of atypical antipsychotics. And initially, this substance was being given to rats in order to look at models of psychosis. But more recently, and perhaps more interestingly, they've also been identified as a potential novel antidepressant target. And the effects in the limbic system are probably particularly pertinent in that context. So this, oh, sorry, just get my slides to move. So as I say, this was an analysis project I've been working on as part of an MRes in order to sort of build up my expertise and explore this data set in a bit more detail than had been done when it was initially collected. So this study is looking at 5 Wistar rats, and they were placed in an open field arena that was familiar to them, sort of circular space that got tether and LFP electrodes in place, and they're sort of free to explore that environment. They recorded in the arena. There's no sort of task or anything. They're just freely exploring. 50 minutes pre-inject and 40 to 90 minutes post-injection. They received after an injection of this 25 NBO8, which is things that have been used traditionally, such as DOI or LSD, or clinical trials psilocybin, which are a lot less selective, or they were giving a vehicle injection as a control. So the Wistar rats that we're using had LFP electrodes in the basolateral amygdala, in 2 sites in the prefrontal cortex and the infralimbic and prelimbic cortex, and also in the dorsal hippocampus in CA1. So far, I've looked at this data using spectral analysis, Cronux toolbox, which I'm going to present next, and then looked at Granger causality using the multivariate Granger causality toolbox. So this is an example of a spectrogram from a single rat, and this is from the LFP electrode in the infralimbic cortex. The reason I've chosen this trace is because, or this brain region rather, is because it's the strongest signals of changes from the 25I injection. So what you see along the bottom is time relative to the injection. The black dotted line is the injection point, and then you've got frequency from 0 to 200 hertz on the y-axis. Following the injection, there's a sudden appearance of a high power, high frequency oscillation. In the different rats, it varies slightly in frequency, but around 125 to 150 hertz, sometimes slightly higher. That appears to come on consistently in infralimbic, prelimbic, and to a lesser extent in the basolateral amygdala. There's also an increase in sort of persistent gamma activity in the sort of high gamma region. And if you look on the spectral analysis, there's also a blackening of the background. So there's a suppression outside of these bands of high power. Now, these high frequency oscillations have previously been reported with some subanesthetic doses of ketamine, and initially reported in the nucleus accumbens, but also found in prefrontal cortex with ketamine as well. I think this is of note, given that ketamine has also proved to be a novel antidepressant and has come into recent clinical use. So part of my interest is whether or not these high frequency oscillations could have in some way mediate an antidepressant effect. For comparison, this is a control spectrogram. It's aligned around the injection time on the same scale. As you can see, it looks very similar to the pre-drug period. And whilst there is some activity in this sort of high frequency oscillation range, it's not with the same degree of power or as much organization, and you haven't got that real blackening of the background.
5 minutes.
Okay. So just going on for a few more minutes. So with a single rat after the injection, this is 30 minutes before, it's just a heat map showing where the rat is, whereas this is 90 minutes after, and you can see that the rats become a lot less mobile, and they also stay towards the edges of the field, showing a strong sort of behavioral effect. If you look at the power spectra, this is what I was talking about on the spectrogram. So this, the blue line is the pre-injection, and you get this increase in power in the high frequency ranges, and a background suppression that seems more global. When we compare this to the control data, especially if you look at the different spectra here, which is the change in power pre and post drug, you can see there's very little change over the 40 minutes of recording in comparison to the drug injection. When we look at the Granger causality, blue line on these plots represents the pre-drug period, red line's early post-drug, and yellow line's late post-drug. What we see is that there's a significant change in the Granger causality from the infralimbic cortex to the basolateral amygdala. So this is a statistical measure of functional connectivity that's saying that there is a information flow, or that the signal in the infralimbic cortex predicts that in the basolateral amygdala, and this appears to spike following 25I administration. And what we see is that there isn't a reciprocal signal going in the other direction. So there's an increase in the directional signal between these two brain regions. There's a few other things going on with the Granger, but nothing with the same intensity of signal. And so we're looking further at this. And the next steps are going to be to explore it with dynamic causal modeling, to try and explore the sort of effective connectivity. So what sort of neurobiological model could underlie this, or how the connections are changing. And just for comparison, if we look at the Granger causality for the vehicle injection, see that although there is this signal from the infralimbic cortex, the basolateral amygdala, there's no particular change over the pre, early, and late injection periods. All right.
That's all. Thank you, Daniel. So I'll open the floor for questions. Yeah. Don't see any questions. Yeah, I do have questions. First, I was wondering whether, so you mentioned that this, that ketamine was doing something similar to what you were seeing. Was it specific to the 140 hertz peak that you were seeing, or was it to other peaks that we were seeing changing once you put the 5-HT2A agonists?
Yes, so the ketamine effectively causes this same peak here from around 120 to 150 hertz.
And so do you know if there's any sort of normal behavioral states that would be associated with that sort of peak that has been seen without any sort of pharmacological manipulations?
Not that I'm aware of. I don't think we've really got a putative physiological explanation for this, or any particular explanation of why it's happening. There are I guess physiological processes that have high frequency oscillations, but not sustained and in that same sort of pattern. So yeah, I think that's something that we're part of the reason we're interested in this.
Yeah, super high. Yeah. It's super intense. You'd expect, if it's doing something like ketamine, you'd expect it'd be doing some type of hallucinogenic situation, right? That's, I guess, the background of this.
Yeah, and obviously this drug will have sort of psychedelic hallucinogenic properties as well as ketamine.
If I may have is there a question? Yeah, I see a question. We have Marima here who asks, how do you think the behavioral inhibition shown in your heat map data plays into the functional connectivity effects? Are they linked at all?
Yeah, I think that's a good question. I think it's difficult to say. I have looked at what happens when you look at movement speed to the power spectra, and in periods where the rat is more stationary, then the changes are more pronounced. So you get further blackening of the background, and you get a greater power of the peak. When the rat is moving, then that's lost. But obviously, there are certainly in this control trace, there are still periods where the rat is relatively stationary. Yet we're not seeing the emergence of the same.
So wait, let me just get that straight. So when the rat was moving around and not being stationary, you've reanalyzed this, and what happened?
So you get effectively just a more pronounced version of the pattern.
More pronounced.
So in these patches here, so you can see there's a break in that. There, it's not particularly obvious from this speed trace, but in general, the breaks in the high frequency oscillations are at times when the rodent is moving.
And the question was also about the functional connectivity. Did you do the same for the functional connectivity?
So I haven't looked at the Granger to see if it's different during those periods. It is something that's being looked at with respect to the dynamic causal modeling to see if there are changes in how things are connected up. That's a good point on looking at the Granger at those times. And I guess from the other thing that we wondered about was to what extent is the sort of clinical properties of these drugs related to the movement change. I think the answer is that actually these the dose that's been given in these experiments is pretty high compared to what we'd use clinically, and so I don't think it's surprising that there's reduced sort of movement. But even in the sort of clinical use, actually, people do stay pretty still in the therapy and sort of put on eye shades, and so in the antidepressant use, it's sort of similar, I guess.
Cool. Thanks. Great question. Great detailed answer. So I had another question. It's a little bit naive, maybe. I was wondering how specific was the agonist that you're using to 5-HT2A?
I don't know in terms of sort of numbers, but it's like a highly specific.
Highly specific.
Highly specific. So I think it's been developed as a radioligand for tracing of these receptors in like PET studies and things.
I see.
Yeah.
Oh wow. Okay.
There is another 25 CN, which is more specific, but this already has pretty high specificity. We have got some potential data on the other drug, which we might look at in comparison.
Cool. Fantastic. All right. Well, thank you. Oh, Yaroslav has a question?
May I? Yeah.
Sure.
So basically, I was wondering how is this increase in this high-frequency power band relates to the multi-unit firing rate? Can it be just like that there's number of spikes goes up in IL, or
yeah,
also
unfortunately,
pushes the power band and pushes the range as well?
Yeah, unfortunately, this was conducted with LFP wires, so we haven't got tetrodes and unit recording. So I'm afraid I don't know the answer to that. I think in terms of, how the frequency, could feasibly be so high physiologically, then yeah, I think you're right. It's it's going to be increased frequency of, it's going to be multiple units spiking in a either sequentially or together, given that each single neuron's not going to be sort of firing at that rate.
Mm-hmm.
Thank you, Daniel. We'll switch to our next speaker. Maybe you can unshare, and we'll have Sean sharing his screen. Thank you very much, Daniel. Next up, we have Sean Martin, who's speaking to us from Trinity College in Dublin. And his talk is on the analysis of interregional connectivity at the single cell level. So take it from here, Sean.
Great. Hi, everyone. I'm Sean, and I'll be presenting some joint work with John Aggleton and Shane O'Mara at Trinity College Dublin and Carnegie University. So yeah, we'll be roughly looking at the probability of recording anatomically connected neurons in multiple interacting brain regions. And the real motivation for this is that there's these amazing, huge data sets being recorded at the moment where they've numerous neurons in multiple brain regions being recorded by Allen Institute, Janiela, Brain. We're talking about it yesterday, that these sort of hero data sets might take decades to analyze, but it's the kind of thing we're thinking about. And really what we think is that the correlations and the patterns, the basis for this should be the structural connectivity. In theory, in some sense, it must be. So that's what we're looking at here. To put the problem quite roughly, if you've got two brain regions and you sample a bunch of neurons randomly in the two, such as these gray ones here, you could get them randomly, what proportion of them would you expect to be connected? So for here, example 7 is connected to G by a direct connection. And so if you scale it up to a bigger network, such as 1,000 neurons in both regions, which is still orders of magnitude less than would be in a real brain, but gets the point across, hopefully, for an illustration. If you were to take, say, 20 randomly here and here, how many might be connected? Or in this kind of network, many different kinds of networks. And so when you look at this and you want to solve this problem, what you need is some kind of recipe of explaining what the connectivity between the two regions you're looking at is. Here, for example, we have a full connectivity matrix from the blue brain model of the mouse neocortex. But it could be anything, any kind of recipe that says how forward connections between the regions are, what recurrent connections are like, what local connections are like. You just need some sort of recipe. And then when you have that, you turn it into a statistical model so that you can say, okay, if we sample X neurons here and Y neurons here, how many of them might be connected to each other and along what kind of paths? So to do this, we look at, say, take this graph here. You want to do some computer simulations of it first for accuracy purposes. So you just literally over and over again, take a handful of neurons on one side randomly and a handful on the other randomly, and you get a distribution by simulating there, and you compare that to the distribution from your statistics. So here, for example, it's taking 20 in both regions, and the probability mass function matches quite well. There's some small differences for sure, but the overall shape is similar, and more importantly, the mean is very similar. You can see that again if you take different numbers of samples. So we take, say, 5 in each region and so on. It matches quite well. And here there's a distinction. The blue line is indicating that these neurons that you sampled randomly have to be directly connected. The orange one is saying that they don't have to be directly connected. They could be, but they can also communicate through one other neuron. And similarly for the green is through 2 other neurons. But the match is quite well overall. And you can do that for different kinds of networks, different kinds of connectivity, different brain regions, just a few samples here, but overall the expectation tends to match quite well. And if you apply that to some real data, for example, well, real is in kind of the best we're looking at the moment, perhaps with the blue brain model again of the mouse neocortex. Here we're just taking primary motor cortex area to the primary somatosensory area associated with lower limbic function. And you take different numbers of samples in those regions. So down here, you might be looking at maybe a tetrode in both. Up here, you're looking at maybe a neuropixels probe in both. And you're just trying to ask the question of how does the proportion of connections grow with the number of samples you're taking. So for direct, it's roughly linear. For them being able to communicate through one other neuron, so 2 synapses, it's roughly exponential. And with 3, nearly everything is connected just because these local connections back here are so vast. And then you can do that for lots of different brain regions. Here's just a few examples. We already mentioned this one just there. You could do that in reverse direction, so somatosensory to motor instead of motor to somatosensory. You could look at primary visual area to lateral visual area, and in the reverse direction. And in this case, we're just taking 79 samples in both of those brain regions, which is the average of a neuropixels probe. So to conclude, the statistical estimate that we have here is fast. It's much faster than simulating these networks. It takes a really long time to have to get these individual neurons and see if they're connected. It's quite accurate. We're in the process of quantifying how accurate. As you saw, there's small differences in the probability mass function. And some of that comes from how many iterations you do, some of it's other factors. We do have a large limitation in that we're not considering topography, by which a neuropixels probe actually is a linear shaft, and you would expect all the neurons you sample to kind of be in a line, whereas we're just taking 79 neurons kind of just randomly everywhere. We're not considering space. So that is a big limitation. And dynamics is also not considered. We're just taking a static network. We're not considering synaptogenesis or anything like that during your recordings. But in the future, we do hope to try and think about how this could be applied to large data sets. One example, just as a kind of to think about, might be, say you noticed a very high correlation between 2 brain regions in these big data sets that people are recording. The neurons tended to spike well together. And this also showed that they're very highly likely to be well connected. Perhaps that's indicative of those 2 regions may be driving each other as opposed to being driven by an external region or by overall oscillations in the brain. But that's obviously for future thinking. But that's me. Thanks very much for listening to the talk. You can find us at these email addresses and websites. I'm happy to take any questions. And thanks again.
Great talk. Thank you, Sean. So we're open for questions. I guess I'll start off. So you said that you set up that algorithm to determine the number of connections effectively. So what are the caveats that you would expect could be taking place? So you're happy to have the same mean number of connections, but what would be possibly going wrong?
Yeah, I think really what's going wrong is kind of to come back to the topography limitation. I think really, if you take a tetrode and you take, say you got 5 neurons that they're all kind of in a small ball, they're not reaching out properly to the rest of the neurons in the brain the same way the 5 we're just taking kind of, we don't have space like the blue brain model or the recipe. You could kind of try to infer space based on doing, like if you take the network and you try and do forces, maybe modeled as like a spring particle system to, maybe guess what space is, but it's not fully in the model. So we're really missing out on space, I think, is the real thing that could be going wrong. Yeah.
Other questions? And you said, so you wanted to infer the number and also the kind of path. What did you mean by that?
Yeah, by kind, I just meant, does it go, is it a direct connection? Is it through one other neuron, say, to help them to finish the communication? Is it through to, we're not considering excitatory or inhibitory. For example, you could set it up with only excitatory neurons in this or inhibitory. By type, I just meant length of path, sorry.
Cool. Yeah, I see. So disynaptic, trisynaptic.
Yeah.
And specifically to this, you were mentioning, I didn't quite get your graph where you were saying if you have disynaptic, we get more connections, and trisynaptic, basically they're all connected. Could you maybe expand on the implications of this?
Yeah, so this is oops. Basically what we're just saying here is that, now this is for, we do have results for like the kind of blue brain model as well. This is more for this kind of toy example. But really what we're just saying is that if you're trying to look for directly connected neurons in these kind of recordings, it's still, if we go to the blue one, it's still going to be pretty low chances. But if you're willing to consider maybe a kind of a lower influence of neurons on each other, then you will see with these, larger recordings that there's a lot of influence between these neurons that you're recording.
Yeah, okay, so if your technique for spotting a connectivity is agnostic as to the number of synaptic jumps beyond 3, you basically should see full connectivity. That's what you're saying?
Yeah. And of course, topography is still, the limitation of space is still there. So it's the one thing we kind of have to look at. But even I think at that, if you're taking 4 synapses, that I think even with space as a consideration, you're still, nearly everything is going to be connected.
Wow.
Or at least as long as there's good connectivity.
It goes pretty quickly. Like, is that for the blue brain or that's for your?
Yeah, that's for blue brain between. It's for this one here. Oops, I'm going the wrong way in my slides. No, it goes really quickly. It didn't expect it.
So the connectivity, the basic connectivity between excitatory neurons in there should be something like 5%, right?
Yeah, but it basically explodes so quickly when you allow, like, even though we were surprised too, and I'm still kind of, to some sense, coming to the ramifications of that, because I set out with this project almost to be like, we're doing great with neuropixels, but it's still not enough. And then I went, whoa, actually, as long as you're not looking for direct, it kind of nearly is enough. It's just the local connections, like even, this is obviously averaging over a lot. These are 150. Each one of these squares is 150 by 150 neurons averaged for their number, like how many of them are connected, the percent. But regardless, these local connections, even as you say, there's not that many of them, but it just explodes when you allow.
That's good. It's kind of surprising.
Yeah, no, I'm still kind of coming to the ramifications of that, to be honest.
We have a question here from Sébastien Maillet, whom I actually know, who says, first, thanks for the great talk. And he says, do you think that your analysis could also help to improve the interpretation of retrograde, anterograde labeling studies with smaller data sets?
Yeah, possibly. It's definitely something we're thinking about. It's really something in mind, especially John Ogleton does a good bit of work in this field. So it's something in mind. I would love to give you something more concrete than that, but I'm not entirely sure, to be fully honest.
And what are the MOP and SSP?
So MOP is primary motor cortex is MOP, and SSPLL is the somatosensory, primary somatosensory area associated with lower limbic function. It's the labels from Blue Brain's own
Institute.
Blue Brain in this case, but yeah, same for Alan, really. Yeah.
That's all my questions. Any other questions from the audience or from the panelists? If not, we would switch to our next speaker. Thank you so much, Sean. So, Jaroslav, I now see your slides. Okay, so hi. So the next, yes, go ahead. Sorry.
Eric, you wanted to introduce her?
Yeah, I wanted to make the full introduction and say your name wrongly. So next up, we have Jaroslav Zich, or Zich, I don't know, from the University of Zurich, who's working with Fridtjof Helmchen, and he's going to talk to us about the mesoscale brain dynamics and how it reorganizes during learning of tactile texture discrimination. Looks very interesting, so take it out.
Yeah, thank you for hosting me here, and it's great follow-up for the previous two talks. Yeah, I'm Jaro, and I'll talk about mesoscale networks and how the connectivity changes during behavior. So our study mainly focused, or my interest was, how does behavior affect this global connectivity, and we use tactile texture discrimination task to assess it in mice. So we don't look only at the microcircuit, at the barrel cortex, but we also try to connect this whole basal ganglia thalamocortical loop, how the sensory information is fed into the motor circuits for mice to engage and complete the task. And we're also motivated by several recent studies, and not so recent as well, but there are just a few examples here, where people found the global brain activity with neuropixels probes, LFPs, multiple LFP probes, during behavior. We used multi-site or high-density fiber photometry for this. So we implant an array of thin optical fibers into the mouse brain. Here's the coronal section where we sample from basal ganglia, thalamus, cortex, and even hippocampus. And this part is implanted in the mouse longitudinally, and it's connected to the via fiber bundle to the wide field microscope. So on the camera, we can record a fluorescence image from the GCAM calcium sensor, which is a good proxy for the multi-unit activity in each brain region. Here's task which mice learn. It's the texture discrimination. So the timeline here is important. The texture is presented to the mouse whiskers. It's a Hedwig's task. At the second number 3, they have to whisk and discriminate between hit and correct, between go and no-go texture. And correct trials would be hit and correct rejection, because for hit, mice have to lick and retrieve a reward, which is given at the second number 6. And for no-go texture, they have to emit any action. And of course, they can also do miss or false alarm trials, which would be error trials. Mice can learn this task very fast. Within a week, they become experts at the criterion of 70% correct trials for us. So here is the video example of expert mouse. So you see texture approaching, and for hit trial, mouse is licking, is retrieving a reward, and for correct rejection trial, it emits any action. What was surprising for me to see, here's the delta F over F of changes of calcium activity across 48 different brain regions in the mouse brain of the same hemisphere. The surprise for me was that the connectivity, the activation across these brain regions is so distributed and exists even in hippocampus and some of the midbrain nuclei. So, but because we look also during learning, we look at which behavioral parameters change. Indeed, we have also changed in some motor parameters. Mice whisk more and lick stronger, so they have a stronger vigor to retrieve their reward. And if we look at the changes in the correlation of these behavioral parameters to calcium signals, we see that the correlation to touch is a Pearson correlation, is increasing in basal ganglia, motor and somatosensory cortex, and ventral medial thalamus, while correlation to licking is decreasing. So the licking for a mouse is not the goal-directed motor command anymore, because they know that the texture which is predicting their reward. What we also see is that the discrimination power, if we simply train the linear discriminator on these calcium traces in this very early period of the texture presentation, from 3 to 3.5 seconds before mice lick on the first time on the water spout, we see that mice can discriminate the hit versus correct rejection trials. Calcium signals can discriminate in barrel cortex, but also in basal ganglia, thalamus, and even hippocampus. This motivated us to look at the functional connectivity. We use the transfer entropy. It's basically a measure, a directed measure in our case, calcium time series, when the source area, the past calcium dynamics in the source area is predictive of the present calcium dynamics in the target area, given that the past of the target is not predictive of its own present. And if we just take the simplest measure of the dysfunctional connectivity across learning, is just simply number of links in this network of regions which we sampled from, we see that the number of connections is increasing throughout learning, from the naive to the expert performance. If we plot the top 8 links in this network, so basically the subset of links which is increasing its connection strength throughout learning, which is shown here in this blue histogram on the left for heat trials, versus the naive connection strength, we could see that throughout learning, mice established not only cortical-cortical communication between motors and somatosensory cortex, which has been previously shown and we would indeed expect it to happen, but also that the information is fed from the somatosensory baron cortex via thalamus back into the cortex. What was also surprising for us to see is that actually these functional networks during the time interval before the first link are very similar for heat and correct rejection trials. And we also looked at the network beyond the somatosensory regions, namely that we looked at the prefrontal regions and some of the posterior regions. We also could recapitulate results with the 12 fiber implants, which means that we have still increase of functional connectivity throughout learning, particularly during the texture presentation time. And we also see globally that medial orbita, frontal and prefrontal cortex became hub regions, because this early texture presentation period, cortical and subcortical regions were feeding information into these regions. And also internal globus pallidus, if we look at the changes in the clustering coefficient, also increase this clustering coefficient in comparison to all the other brain regions during this early texture presentation period. And clustering coefficient is basically the probability, if you send outgoing links, that your target regions which receive a link from you also establish a link, no matter in which direction. So this is some sort of a hallmark of the pathways related, like metascale computation for us. That's why we also related to clustering coefficient. You could also check details of this work in the bioarc. And here are the acknowledgements of the group of people I worked with: is Professor Fritjof Henkin, Alexei, and Mel. Thank you for your attention, and I'm open for questions.
Thank you for that's quite impressive, Jaroslav. There's quite a lot of stuff in there. I'm sure there are a lot of questions. I have one maybe to start. I'm also interested, I've been really interested in trying to figure out what changes during learning, but also what causes the changes that are bringing about the expert state. And I've been focusing on the idea that there should be some type of learning signal or lack of equilibrium that is present during learning, but that would be gone once you're in the expert state. You see? Do you see what I mean? So it's a slight, I think you've analyzed a lot your data from going to naive to expert, what changes. But in my view, there should be something in between that is present in between the two that vanishes once you're in expert, because you finally, you've reached expert state, so you don't have to learn anymore. Have you looked at your data this way?
Yes. So the beginning of this project was I was also interested in by this idea of Darwinian learning, right? That the neural circuit, if we speak about somatosensory, even like a microcircuit, that it would first during learning recruit multiple neurons of the similar tuning, be it auditory or visual, right? And then after the expert performance, it would drop out all those which have a tuning which is not relevant for this particular task. For example, it's the tuning for discrimination of 2 and 4 kilohertz, right? So in this case, the local circuit is further expanding in the number of neurons it recruits for making the task, and then it's collapsing, it's reducing again. And in my case, I was also curious about it, but first the analysis for this transfer entropy requires many trials because it does some sort of the bootstrap, because I end up with p-values for its transfer entropy, which is actually a link, not the magnitude of the transfer entropy itself. So my time step for the analysis is relatively coarse. It's only a day, right? I could take a day of the learning day itself. What I saw in some subset of mice is that indeed they first increased and some of them decreased their functional connectivity when they became overtrained, but that was only a trend. I cannot say it's like actually present in all mice.
Very interesting. Maybe to follow up on this, and since you mentioned, and maybe to echo Sean's talk, how did you actually compute? So you said you used a transfer entropy to determine the number of links, but I was a bit confused as to how you extracted the number of links using transfer entropy.
Not number of links. So for each region, I've calculated transfer entropy in the moving window, so the which maximize transfer entropy for the present time bin, and then the past 5 time bins were used for each region A and B, right? So A, B being the source and B being a target. So the source could transfer the information within this 5 past time steps to the target. And but basically 90% of links came from the last time step only. So the closest to the present time step of the target. And I did not extract the number of links, but if this influence is statistically significant, which is determined by the shuffling of this past time bin within the same brain region source A, with the same type of trial, hit or correct rejection, but across the different trial, right? So once it's shuffled, you destroy this momentarily relationship between the time series, calcium time series, which you have. And then if it's shuffled and transfer entropy drops, then the p-value is below 1%, and that's the binary threshold for me of the effectively establishing the link. So it's a one link. And then I do all permutations for all possible combinations of brain regions, and then I get the adjacency or connectivity matrix that is still binary. And then I have free time samples, which is happening between the picture stops and mouse has a first click. And from this binary matrix, I get a weighted matrix of the connections, because some can be repeated, right? Basically, and some may not be in steps of 0.3.
That sounds good. Okay. Yeah. So basically, you count the number of inter areas that have this drop in p-value when you shuffle the data. Okay.
Yeah. So only significant ones I count as links. Yeah, which drop in the.
And then you scale them by the amplitude. Yeah. You had at the beginning, you had this. You showed very quickly that you said this was the activity across the different regions, and it was flashing. It was a bit of a movie, right?
Yeah.
Right there. So, like, how come we're seeing the data in those circles? What's happening there? What do we see?
So these are multiple fibers. They are connected to the fiber implant, which is chronically implanted in the mouse. So the increases in fluorescence relative to the baseline are shown in the middle row, and the maximum when you see this white is about 5% delta 4F change. And this is the changes of calcium dynamics, which are related to the multi-unit activity of neurons sitting directly below the fiber tip. Each fiber is 120 micron in diameter. So from some of the Monte Carlo simulations of other people, the fluorescence signal drops by a factor of 100 on the diameter of the fiber down. So it's basically, I'm expecting a volume of 100 by 100 by 100 micron of the brain tissue contributing to this signal. So it's, if you want me to end up with estimate of number of neurons, that
no.
that would be hard because you referred to Sean's talk. That still would be hard to guess, but I think it also depends on the brain region. I think it's in around 100.
Yeah. Okay. Cool. And I guess my last question, if there's not any, I don't see any other questions here. Is, well, you've shown quite a lot of changes. Maybe and you've discussed a few summary statistics, and the hub stuff look really interesting. But did you find particular changes that you found were particularly interesting, or that were particularly striking your attention, that you were in the lab that you thought, wow, this is really surprising? Diving into that data.
Yeah, I think what was surprising for me is to see this connectivity matrices for 48 channel mice, and see that you can see that here this red increase in the connectivity for prefrontal cingulate medial orbitofrontal cortex is kind of means that multiple brain regions are giving the inputs at this early period. It's kind of, you can think of it as the bottom-up flow of the information. So that was impressive for me to see. I was even thinking first it's some sort of a bug.
Yeah, it is very interesting. And how did you create those hierarchical graphs?
These plots are basically the top 1 percentile of the strongest links from this matrix here, just to show the top strongest changes in the connectivity, which happen from the baseline period where the texture did not approach yet the whisker, and for the period of detection, this early texture discrimination period.
Oh, in a representation where the distance scales with the correlation.
In the representation.
I mean, we see that some centers are farther apart than others.
On this plot.
On this plot, right.
Yeah, it's a force-directed placement. Yeah, so it kind of tries to pull the nodes which have similar patterns, similarly projecting into each other, or supposedly similarly projecting to each other closer, while others further apart. Yeah.
Got it. All right. Well, with that, I think I'll close the stage. Say thank you to you again, Yaroslav, and to all the panelists. And well, thank you, Elena, for covering up the backstage. And let's enjoy the rest of the conference.
Thanks, guys. Thanks very much. Thanks, Richard.
Please post questions on the Q&A panel. The talk format is discussion-based, so please feel free to ask any kind of questions. There are no such thing as dumb questions. Vote for your favorite questions in the Q&A panel, and those will be asked first. If you would like to ask your questions on screen, please put the on screen in your question box, and we'll invite you to join us. You can chat with the panelists or everyone here. So with that introduction, I'm going to ask our first speaker to come online. He is James Goodman. He's a postdoctoral scientist at the German Primate Center. The title of his talk is Group-Specific Dynamics Are Not Shared Between Action and Observation in the Frontoparietal Cortical Grasping Network.
Thank you.
Welcome, James.
All right, let's get started. In our lab, we are interested in hand movements and the frontoparietal cortical grasping network that enables them. Work from our lab and others has demonstrated that this network performs the critical task of taking visual information about objects and gradually converting that into a movement plan as those signals progress from AIP to F5 to M1. However, another major line of research in this network involves how neurons in these areas respond during the mere observation of action, responses which are thought to indicate a sort of motor simulation. We sought to apply population analysis to this thread of grasping research, which to date has been scarce. To this end, we trained monkeys to perform a grasping task in two different contexts, memory guided, which is in the dark, and visually guided in the light. We also trained animals to hold still and watch a human experimenter grasp the same object. We presented a wide variety of different objects to elicit different grips, and we used floating microelectrode arrays to record from the three major nodes of the frontoparietal network. And the neurons we recorded across these areas exhibited a wide variety of different preferences for action and observation. Shown here are peri-event time histograms of two example neurons showing high selectivity for one or the other, representing the range of responses we see across these areas. And the first thing we do to try and get a handle on this is we use just principal components analysis to visualize the activity of this diverse population of neurons in state space. We see when we do this that AIP exhibits robust activity across all three tasks. And as we progress further along the visuomotor hierarchy, we gradually see that the red observation component shrivels away, consistent with what we would expect. While these plots tend to highlight the differences between these tasks, we also see, when using demixed principal components analysis, a substantial overlapping component between action and observation in F5 and AIP. However, an ideal test of motor simulation should come in the form of common object representations rather than merely a common activity pattern that doesn't vary with any of the experimental conditions. Indeed, we should expect that if we classify grip type using neural activity recorded during movement, we should be able to use the same readout to determine those same grips during observation, and vice versa. So we run a cross-classification analysis in this vein, comparing each pair of tasks at all possible pairs of epochs and timings. And we end up with these heat maps detailing each and every one of those pairwise cross-classification performances. This is a lot to take in, so I want to draw your attention to two things. One, how an M1 cross-classification is high between the two grasping tasks during the movement itself, but not anywhere else. And two, how an F5 cross-classification is high between different tasks and epochs generally, except when you consider when movement is occurring during the observation task. In general, across all areas and in two animals, we fail to see high cross-classification accuracy when comparing action and observation. But okay, this is all a very representational framework. What if we consider a complementary dynamical systems framework of neural activity? One where we, for example, use linear regression to fit the firing rates and their derivatives in state space, and ultimately determine how well the dynamics of one task explain activity in the other. How similar are action and observation in this framework? In fact, they remain quite dissimilar. If we build the same pairwise heat maps that we did for classification, only this time using R² instead of classification accuracy as our measure of goodness of fit, we see that in both M1 and F5, there is high congruence between the two grasp tasks, but again, poor fits when comparing action and observation. Again, this pattern holds across cortical areas and animals. In conclusion, we fail to see any indication of grip congruence between action and observation activity, whether we used a representational or a dynamical systems perspective. This would seem to argue for at least one of three possibilities, one where there is simply no motor simulation being recruited in this context, one where the shared representation of action concerns features that are more abstract than the specific grips, and one where there exists some distinct simulation subspace which avoids, for example, executing unwanted movements. And with that, I'd like to thank you for your time and invite you to ask any questions.
Thank you, James. That was on point and beautiful graphics, I have to say. And we are waiting for questions to show up on the Q&A side of things. I would, if you don't mind, I would like to start with a couple of questions.
Sure.
One is the idea of, you were talking about your model. Are these primates already trained in these tasks, or is this just the first time that they're interacting with these groups?
They are well trained in this task. We're not looking at anything resembling, say, motor learning with this process. Yeah, they're hyper trained on these tasks. They're grasping these objects pretty much every day.
So would you expect that if this was more about learning aspects of it, that you may be able to see more of a congruence between action and observation?
Perhaps. Admittedly, we don't really have the data to assess that claim. It was one of the lines of study that we had been interested in pursuing down the line, but as of now, we don't really have the data to say yes or no.
I see. Okay, all right. So another question would be thinking about it from what other regions would you perhaps might expect this to be happening in, since you're not seeing this congruence between action and observation in these regions that you're looking at.
Right. It's tricky, right? I mean, we might expect to see it, for example, I mean, the whole idea of this is that it should be related to motor control in general, right? Because otherwise it's hard to make a claim that it's a replay of a motor plan. I guess the answer to that question is this is kind of most of the areas that we should expect this to be in, right? Like these are, to call back to previous literature, these are the classical areas that show mirror neuronal activity, which is very strongly related to this idea of motor simulation.
Mm-hmm. Right. Okay. Other panelists, you're welcome to ask questions as well. This doesn't just have to be a one-on-one discussion, so feel free to unmute yourself if you have questions too.
Yeah, I'd actually like to ask a question. So you showed in those correlation matrix plots, you showed high correlation during the wait period, but not the move period. What's the interpretation of the correlation during the wait period?
So the idea is that this would reflect a common visual or memory period kind of activation, right? Because I didn't go over the details of the task, but what happens is a light shines, the animal sees the object, and from here things diverge, but the long and short of it is after a certain delay, the animal then reaches for and grasps the object. So from the time that light shines and the animal sees the object up until they're grasping, that's what this wait period is. And basically it's what the animal sees, which should be consistent. Oops. It should be consistent across all the different tasks. With the exception of the memory guided condition where the light eventually turns off, but then you have what we might call a memory, a memory signal, a persistent memory of the object.
Great.
So,
go ahead, sorry.
So during this wait period, how do you dissociate between the visual or like the sensory experience and motor preparatory signals?
Yeah, that is certainly a very tricky problem, and colleagues in the lab, notably Benjamin Dang and students that he is working with, are looking at methods to do precisely that.
Okay, we have one question coming online. Saloni Sharma, would you like to come online to ask the question?
Hi, sorry, that took me unaware. So I was just wondering because in the mirror neuron research, these strictly congruent mirror neurons have been well shown in F5, where they really seem to respond exactly to the grip that they well observe. How do you reconcile what you find with this previous so far established research?
So ultimately, what I mean, the way I reconcile it is essentially how congruent are these grip representations really? You know, when we look in the mirror neuronal research, what we find are a bunch of example neurons, and normally the very highest performing ones. Generally, these neurons are not very numerous. They comprise a very small fraction of what's going on in the population.
Mm-hmm.
And so I think what's happening is, you know, very early on, I showed, you know, 2 peri-event time histograms that show the range of responses that we see across the population. What I think is happening is you're seeing a tail end of the distribution where perhaps by chance you're seeing high congruence across the different conditions. But if you look at the whole population level, if you look at what's happening across the network, the level of congruence between the observed and the performed action is not really so high.
Okay. I just have one other question, if I can still ask.
Please, yes.
So I'm curious about your, well, one of your suggestions as your conclusion, where you say there is no motor simulation in this context. So I'm just going to go very broad here and say, are you saying that mirror neurons don't exist, or that there is like, yeah, or like, I was just wondering what exactly you mean by that?
That is precisely what would be the case if this were indeed that hypothesis, if that hypothesis were indeed true.
But you do, there is, like you can see in the F5 neurons, especially, I guess more medially, that they do seem to respond when there is a visual during observation, right? It's not like the response isn't there. So
it's not that there isn't a response. And again, I mentioned that there is a component of activity that is related, that is consistent across all 3 tasks. I didn't show it explicitly, but if you look in F5 and AIP for that matter, there is a common component where all the conditions line up and you see this general waxing and waning of activity associated with that.
Yeah.
It's tricky to base assumptions of a motor simulation based on just that.
Yeah.
Because it could be any number of other factors that are common across the different tasks, which is many in this case.
Yeah, and the other thing is, I guess, I don't know if, because I guess you were restricting yourself to the convexity, so F5C, but if you go to the bank where F5A is, you might find more of a congruence as well, because F5A really seems to have both motor and visual responses, while F5C doesn't, like the percentage of mirror neurons, let's say, is lower. But that's just what I'm thinking. Yeah, I don't know if bank is as accessible or easily accessible as convexity.
Yeah, it's a possibility. One thing I will note is that for this talk, I did show 2 different arrays in F5 separately.
Yeah.
So I've been looking at trying to figure out differences between the F5 arrays.
Yeah.
You may have noticed that there may have been a slight difference between the F5 arrays when I looked at, you know, population state space. The tricky thing is we're not finding consistent effects that are consistent across animals.
Oh.
So the trend we see in this animal is the opposite of what we see in the other animals.
Right. And how do you exactly know where to place your arrays? It's just anatomically, you're not doing any fMRI or anything.
Admittedly, I'm fairly new to this group, so I'm not entirely certain of the precise procedures. I believe it is guided first by an imaging study to try and figure out where to put the arrays, to formulate a surgical plan, and then you know, we enact that plan.
Okay. Okay. Thanks so much. Thanks.
Thank you so much for the questions, Saloni, and thank you to James for a lovely discussion. I think it's time to move on to our next speaker. Perhaps if you will unshare your screen.
Okay.
Thank you. Our next speaker is Lycia Munoz. She is a PhD student at the Auckland Bioengineering Institute, and her title is A Spiking Neural Network Model of the Motor Cortex. Welcome, Lycia.
Okay, hi everyone. Just checking you can see my slides. Hi everyone, my name's Lycia Haggie and I'm a PhD student at Auckland, as Kirthi said. And today I'll be discussing my spiking neural network model of the motor cortex. So most of us make movements really easily and usually without thinking about it too much. But motor control involves a complex feed-forward and feedback system between the brain and the muscles. This has usually been approached from a neuroscience perspective, which ignores the properties of the end effectors, or from a biomechanical perspective that doesn't look at how activation is generated from the nervous system. So for my PhD, I'm looking at how neural populations in the motor cortex can generate the activity to produce movements, particularly during arm reaching. So I started by modeling the behavior of neurons using a modified leaky integrate and fire equation with Hodgkin-Huxley currents. These equations and parameters are mainly based on the work of Esser et al. The threshold of action potentials varied depending on the state of the neuron, and the effect of 4 different neurotransmitters, including AMPA, GABA-A, GABA-B, and NMDA, were included to contribute to the synaptic current. Neuron types such as excitatory, inhibitory, and pyramidal and thalamic cells were incorporated with different parameters derived from physiological experiments. Single synapses resulted in excitatory postsynaptic potentials or inhibitory postsynaptic potentials, which summate to produce the action potentials. So the motor cortex receives input activity from other regions of the brain, such as the somatosensory premotor areas, as well as the thalamus. And these areas were modeled as spontaneously firing neuron populations, which served as an input to the model. So this is the overall circuit diagram of my motor cortex neural network model, with each of these rectangles representing a population of neurons. It's organized into a layered structure of layer 2, 3, 5, and layer 6. And the input areas are over here on the left. So they're connected by lines. The dashed lines represent slower neurotransmitter behavior, and the thicker lines mean stronger weights and probabilities of connections. So this was all implemented using the Brine2 neural network simulator in Python. So looking at some output of the model, the mean membrane potentials in the model ranged from between -48 millivolts and -75 millivolts, with inhibitory neuron membrane potentials being quite significantly higher than excitatory neurons. The mean spontaneous firing rates of excitatory neurons ranged from 1.44 Hertz to 10.35 Hertz, with inhibitory neurons having significantly higher firing rates, some reaching as high as 36 Hertz. And this may have contributed to the higher mean membrane potentials in the model. So this raster plot shows the overall spiking behavior of neurons over 1.2 seconds, and we observe a much higher firing rate in the inhibitory groups. So from my understanding, inhibitory firing rates can be observably higher than excitatory, but probably not as significantly higher as seen in my model. This is a graph of the interspiking intervals, and it shows that the shorter ISI times are predominant. So currently I'm in the process of parameter testing and investigating the sensitivity of the model to different parameter values. I also hope to look at the impact of scale and the number of neurons in the network, and later on explore how the network responds to a simulated TMS pulse. And hopefully we can replicate some experimental results as well. So eventually I hope to use this model to drive a musculoskeletal model and be able to test different theories of motor control. So thanks so much for listening, and I welcome any questions.
Thank you, Lycia. I have a quick question. So this is your modeling, the paper you referred to, is this mainly looking at human? Where did your data come from for your modeling aspects?
Yeah, so it mainly came from the paper that modeled the human motor cortex.
Right, a human, okay, great, thank you. Any questions from the panelists to get started? while we're awaiting questions in other areas?
Yeah, I have one question. Sure. I'm curious, so what do you think is the output of this model? Is it layer 5 pyramidal tract neuron outputs, or how do you think about exactly what the output is? Because I guess there's multiple different paths that these signals could be going through, right?
Yeah, I was gonna look at layer 5 pyramidal because they're mainly the ones that contribute to the corticospinal tract and then synapse onto the spinal cord and the muscles. So the output will mainly be from layer 5.
Very cool. I had one question. You were mentioning in your data slide that your inhibitory, the IPSPs you were seeing were actually higher than than would have been predicted. So why do you think, what are some speculation and explanations for that result?
I think I've got quite strong like synapses and probabilities in the inhibitory ones. So I think it just needs some like fine tuning to get that excitatory inhibitory balance right in the model. Yeah.
Okay. And the other part I noticed, so I work with the mouse model system, so know very little about the human side of it. I noticed that you had lots of different layers, but not necessarily layer 4. I was curious about, is this a species difference?
Yeah, I think in the human motor cortex, layer 4 is quite like thin, so it's not a very distinct layer.
Okay.
Yeah.
Yeah, in the mouse field, it's beginning to become a bigger deal. People used to think that the mice, especially the M1, the primary motor cortex area, didn't really have a layer 4. It was all supposed to be 2, 3, 5, and 6. And now with beautiful genetic tracing studies, people are beginning to find out that there is actually thalamocortical input directly into M1. And there are PV neurons, paravalbumin positive inhibitor neurons that connect directly back into the striatum. Again, something that you wouldn't necessarily expect. So these are long range interneurons that are doing this specifically in M1. This is not true for all other regions, but in mouse motor system, that seems to be a thing. So I was just curious about it.
Yeah.
We have a question following on Cameron's question. How do you decode the information from the output layer?
Decoding the information from the output layer. I'm hoping to maybe activate a population of neurons and then that generate like an MEP, kind of like in a TMS simulation. So if you activate a population of neurons, then it can generate like a motor twitch. So it'll be from that kind of high frequency output of the layer 5 pyramidal neurons.
Great. Thank you. Any other questions from anybody else? We can always come back for a group discussion towards the end as well. So thank you, Lycia.
Okay, thank you so much.
If you stop on sharing, we can start our next talk. There we go. Our last speaker for the session is Cameron Clayton. Cameron is a 6th-year PhD student in the Speech and Hearing Bioscience and Technology program at Harvard University. He is an NSF GRFP fellow. Congrats, Cameron. His research in Dr. Daniel Pauly's lab at Massachusetts Eye and Ear focuses on the role of auditory cortex circuits in active listening. So the title of his talk is Auditory Corticothalamic Neurons Are Recruited by Motor Preparatory Inputs. Welcome, Cameron.
Thank you, Kirti, and thanks for organizing this. I'm going to maybe switch gears and now talk about motor signals and sensory cortex. Where this project began, though, was we were thinking about the corticothalamic pathway. So layer 6 corticothalamic neurons are the single largest component of the corticofugal projection system and one of the largest classes of projection neuron in the brain. Yet there is no clear role for them in perception and behavior as of yet. But recent advances in optogenetic approaches to activate and silence these neurons has inspired new hypotheses about their role in sensory gain control and predictive coding. So where our work began was starting in auditory cortex, artificially activating these cells with a brief 1 millisecond laser pulse. And what we observed was an alternating wave of enhanced and suppressed spiking that persisted for 100 milliseconds after laser offset. And so that motivated us to ask this question: what extrasensory inputs could serve the role of the laser pulse and activate these cells to modulate cortical sound processing? The outputs of these cells are very well defined within the local circuit, but the inputs schematized here are more mysterious. So one clue that that got us started was thinking about cortical gene expression profiles. And so if we look across all cortical cell types, shown here as these little clusters, we see that L6CTs are virtually unique in the expression of FOXP2. We found this curious because this is a protein that's highly expressed in motor control nuclei and has been implicated in sensory motor processing. So we took that and we went out on a limb and we asked whether motor actions or internal motor corollary inputs which precede movement might be a naturally occurring source of this extrasensory input and basically anticipate or occur around the time of movement triggered sounds to modulate auditory processing. And we use two methods to monitor activity in this cell type during behavior. The first is 2-photon imaging, where we can image hundreds of neurons at 700 microns below the cortical surface because of a specific Cree line. And then for millisecond resolution, we turn to antidromic phototagging, where we express a channel rhodopsin virus in auditory cortex and then activate the axon terminals of these cells in the thalamus. And this leads to a really low jitter action potential that arrives at the L6CT soma. So really briefly, and we can come back to this slide if anyone has questions, we just needed a very simple task to look at this. And so we turned to a just simple licking behavior where the mouse licks a spout to receive a reward. And incidental to that reward, there's sometimes sound and sometimes not. But both the sound and reward only happen with 50% probability. So today I'm just going to be looking at trials where the animal is just licking, there's no sound and no reward. So what does it look like when we look at these neurons? This is an example field of view of the L6CTs. In this neurogram, every row is a neuron, and they're all aligned to the time of lick bout onset. So we can see that there's a lot of activity in the L6CTs before and around the time that movement begins. And that's just sort of summarized here. But we don't see this when we look at upper layer pyramidal neurons. So in separate mice, we looked at layer 2, 3, and really saw something quite divergent from what we saw in layer 6. So to get a better sense of the time course of this activity, we turned to single unit recordings, and again, the antidromic phototagging approach, and basically confirmed with another method this sort of motor related activity in auditory cortex that we had seen with 2-photon imaging. But what's really nice about this approach is that we can put the activity of L6CTs in context, because at the same time we're recording from neurons all throughout the cortical column, and we can also look at putative, inhibitory, and excitatory neurons using their wave shape. So when we looked across all recorded neurons, we observed something similar to what we might have expected from those previous, from what I had previously showed you, which is that we see the earliest and strongest activation in layer 6. And there's some motor related activity in layer 5, but there doesn't seem to be as much in layer 4, layer 2, 3. And one interesting question from previous work on this topic is that there's good work from David Schneider showing this direct projection from motor cortex onto local inhibitory neurons in auditory cortex. So logically, we wondered if these signals that we were observing in layer 6 excitatory neurons, the layer 6 corticothalamic neurons, were happening with a similar latency. And so what you can see here is that basically the activation of L6 RS and FS units seems to be basically overlapping in time. So finally, we wanted to understand, at least in a first pass, what anatomical inputs might imbue these response properties. And so we turned to monosynaptic rabies tracing. And for now, I'm just going to say that the L6CTs are in pink here, and all the green cells are the monosynaptic inputs from throughout the brain. And the biggest motor-related input which we observed was from the globus pallidus and output nucleus of the basal ganglia. And so with that, I just want to conclude by saying that we see motor-related activity in L6 CTs prior to orofacial movements that crucially, and we can discuss this, trigger sound and reward. They seem to occur with a similar latency to putative inhibitory neurons, and they may be supported by inputs from the basal ganglia. And so with that, thank you.
Thank you, Cameron. We have a lot of time for discussion, so I'm happy to take questions in the Q&A part. I could start off a little bit by asking, do you think I might have missed this part, do you think the layer issue where you're only seeing most activation in the deeper layers, is it related to the Cree line that you're using? Is it actually expressed very well in the other layers that you're looking at?
I'm not completely sure if I understand your question. Are you asking if like the Cree line is influencing our observations?
Correct.
Yeah.
Yes.
So what I can say is that all of these recordings happened in NTSR1 Cree mice. So yes, there is a chance that could occur. And obviously we didn't observe this activity in the Thai1 GCamp mouse which we recorded from. But as I said, we are recording from non-Cree expressing neurons as well. And it's less clear they show very similar sensory response properties as we would expect. So that sort of, at least at a first pass, convinces us that there's not some issue with the Cree line in general.
Okay. And one other question is in your early part of when you were introducing the paradigm, are the mice actually on a ball or moving in any way, or is it just stationary and just the lick spout?
So they're in a cradle, so they're not moving at all. Okay. In separate experiments that I didn't talk about here, we had them run on a ball instead to see if that also led to this pre-motor or just motor-related activity before they started running. But yeah, interestingly, we didn't see in that case this similar motor-related activation, which made us really think about whether or not what we're seeing is due to sort of like the predictive consequences of this action, which is namely triggering sound and reward.
Great. Okay, thank you. We have a question from Gonzalo. Hi, Gonzalo. Very nice work. I was wondering if the licking related activation is modulated by the frequency tuning or sound response properties of the L6 neurons.
So we looked at that. So far from what we've seen, there isn't really a clear relationship. It's not like the motor activated cells and the sensory activated cells are distinct populations. It's more of like a Venn diagram where there's some cells that show this mixed encoding of sound and reward, sound and movement.
All right, thank you. Any questions from the panelists?
I suppose I have one. So you used the term corollary discharge to refer to what's going on here. Is there, for example, some evidence of a computation of some kind of expectation or other, or?
So I don't think this work is the best to address this because we really didn't test causally the role of these this activity in suppressing auditory cortex activity. But the idea from previous work, from David Schneider's work, is that these motor corollary inputs from M2 suppress the sounds of, say, your feet as you're walking in the forest. And so he's really nicely shown that if you pair the speed of an animal running on a treadmill with the repetition rate of a tone, there's a really specific plasticity in auditory cortex for that tone when the animal is running, namely the activity in response to that tone goes way down. So that's sort of like what's in the back of our mind is thinking about how this circuit that we've described with layer 6 might interface with that direct projection from motor cortex onto inhibitory neurons, or whether this is an intermediary between them.
We have one more question coming in. Neurons from which layer of M2 are projecting to layer 6 CT?
So We did, I don't think we really looked specifically at layer. My guess is that it was somewhere around layer 5-ish. But yeah, I really can't comment on that specifically. And I think one other real important caveat of our work is that obviously there's a lot of things you have to consider with monosynaptic rabies tracing. It's possible that the virus just wasn't as efficient at reaching motor cortex. So that's definitely something that we're considering and taking these results with a grain of salt. But yeah, definitely the strongest input that we observed was from the globus pallidus.
Very good. I have another question. Which side of the brain are you recording from?
Yeah, we're recording from the right side of the brain.
Okay, you're probably the second physiologist I've talked to that has not been recording from the left side of the brain. So I'm curious because we work on it on cellular plasticity and somatosensory cortex plasticity, and from a cellular point of view, we're seeing hemisphere differences in our mouse system using up retrieval behavior. So I'm always curious when physiologists are talking about recordings on which side that they're looking at. So
yes, yeah.
Right side, interesting. Okay.
Many times it can seem completely arbitrary which side we pick, and to confess for this work, it is. But there do seem to be interesting differences between right and left auditory cortex. And there's the work of Heisel Oviedo and also Bianca Jones-Marlin.
Correct. Yes. From the maternal pup retrieval perspective, all of those kind of studies, the left hemisphere rules for these things. But in our work with somatosensory cortex, we're seeing that it's individual animal dependent variability. So it's not really always one versus the other. But so since you're recording from the auditory cortex, I was curious. Yeah. We have another question. Do the L6 CT neurons show persistent activity patterns to any degree?
I guess I'm not completely sure what that, I mean, I wouldn't say that they show just like tonic activity. What's much more common, I think, are these patterns of ramping around the time of licking, or just basically like brief activation after sensory stimulus.
Mehmet, you're welcome to come online to continue the discussion if you like with Cameron. We do have a couple more minutes. So he may be talking about time range in your activity patterns, but.
Yeah, so I mean, what we can say is that basically, and I don't have the figure here, but the activity when we look instead at activity aligned to look about offset, activity of the L6 CTs seems to basically persist throughout the lick bout when we look at it with EFIS, which is the method that we trust the most to look at sort of questions of time.
Okay, great, thank you. This may be a question for all three of you. I was curious about your first introductory slide, more or less, about, I think it's a provocative idea to think about CT not having much of a function. The idea that maybe we need to figure out what it's actually doing. So I was curious, across species, from human or primates and from rodent studies, how do the rest of the panelists think about this? I find it provocative, right? The massive feedback system with no clear role in integrative brain function and behavior. It's there, we all see it, right? So what is it actually doing? It's a great question, so.
Yeah, I must admit, I'm not terribly well-versed in the problem, so.
Yeah, my understanding of the thalamus is that it's kind of like a relay station that lots of signals come out of, but like no one really knows what it does. So I just like a lot of cells.
So I think, yeah, I think one really important caveat here is that in contrast to maybe the thalamus that you're dealing with in motor cortex or these higher order regions of cortex. This is sensory cortex. So yes, it is just a relay. Well, sort of. But the idea is that these corticothalamic projections might be a way for cortex to actually tune its own inputs And the idea is that if you could do that, and what's really made people excited about these cells is that perhaps you could attend to a specific sensory feature or another, basically just by adjusting the activity of these cells through some sort of long-range input. So our work sort of is like a very first pass at getting it, something like that.
I would agree with you. From a development perspective, that's how I tend to view the inputs coming in from the corticothalamic into the thalamic side, as a way of the cortex manipulating its own inputs as the experience continues through development into adulthood. And in a lot of neurological disorders, like the one that we study, Rett syndrome, we think that that could be a process that's being affected, where there is a disconnect between the cortex and the thalamus. And so there's no more fine tuning of the input that's coming in, which could lead to sensory processing deficits or over expression of these phenotypes. So. But the relay station, the reason we all laugh, Lycia, is because I think this is a very controversial topic. It's become a controversial topic. The others can chime in about it being called just a relay station. I think there are even reviews that call it is not just a relay station, and people are beginning to, and the opposite could be said for the cortex, where cortex is now considered a cover for all the important stuff. It's just a warmer to keep everything underneath nice and safe and happy. So, you know, it's just conversations between different groups that study particular aspects of the brain, right? So,
one other way.
Oh no, you go, Cameron.
One other way you could view these corticothalamic projections as would be as modulating the output of the thalamus to other areas like the striatum. And so it would be a way for cortical signals that maybe happen over a longer time scale or have more contextual information to influence lower, more fine time scale processing.
We have one more question from Mehmet. What do you think the basal ganglia inputs to the L6-CT may be doing?
So the reason that we were so interested when we saw this basal ganglia input is because there's some previous work in primate basal ganglia, specifically in, I guess, the equivalent of the globus pallidus, that basically they see similar ramping activity before self-initiated movements. And so we sort of are inspired to connect our work to that, but I think there's definitely a lot more to be done to specifically manipulate that pathway, which turns out to be very difficult to do technically.
Very cool. Yeah, I would love to see this kind of a system for as an age range issue. If you are able to see, again, what age are your mice when you're doing these?
They're around 8 to 12 weeks. So
8 to 12 weeks, yeah. So it would be very interesting to see as they're learning the leg spout movement and how they are gating and how they are changing their responses to this activity.
Yes.
So, yeah, it's very cool. Any other questions? No, we're not seeing anything anywhere else. We are good. All right. Okay. We are almost on time before we get kicked out. So this is a great time. Oh, Mehmet has one more question. Are there any differences in the dendritic input patterns of M2 versus BG?
So at least what we've seen from looking at like the Allen Brain Atlas with all their injection stuff, we see actually fairly weak M2 input onto A1. And so it seems to target more higher order auditory cortex. But we definitely see axon terminals from the basal ganglia in both layer 1 and layer 6. So that was a nice resource to be able to refer back to.
Okay. Perhaps if Mehmet has more questions, he can find Cameron on his Twitter account and continue the discussion further, and perhaps the bioarchive, right? So we can actually post comments based on the bioarchive. So that would be fantastic to do. So thank you all very much. This was a lovely discussion. I learned a lot from the different systems, and thanks for this interactive session. I hope you had a great time, and we are almost out of time. So thank you, Richard, for hosting everything.
All right, thank you. Thank you for giving me the opportunity to talk. And today I'm going to talk to you a little bit about these specific cells in the hypothalamus and how they're regulated in the stress response. Essential function of the nervous system is to organize physiological and behavioral responses in the face of stress. However, we know that overactivation of these circuits can be damaging. Chronic stress increases the risk of developing mood and anxiety disorders. And conversely, we've shown that, or we know that there's abnormal activation of the stress response in both major depression and PTSD. Stress can also lead to cellular morphology, suggesting some kind of damage, especially in areas that seem to be more sensitive to stress, like the hippocampus shown here. So thus, characterizing the underlying neural circuits is essential to understand how the brain regulates the stress response and how this may become dysregulated in psychiatric illness. So the kind of starting point, stress axis, which I'm going to be talking about, is this region in the hypothalamus called the paraventricular nucleus, or PVN. And these CRH positive cells in the PVN are necessary and sufficient to cause a cascade of events after a stressful stimulus, triggering the release of these chemicals called corticosteroids, which cause the downstream effects of stress, including some of the negative ones across the body. So understanding how these CRH cells are regulated and where they get their information from is very important to understand the regulation of the stress response. One candidate region for regulating the CRH cells is the hippocampus, specifically the ventral hippocampus, which is known for its role in anxiety and emotion. However, it's not known how or when the hippocampus might be exerting its influence over CRH cell activity. One older hypothesis is based on lesion studies of the ventral hippocampus. So these studies suggest that ventral hippocampus negatively regulates CRH cell activity and CORT. So here you're looking at an experiment with a lesioned ventral hippocampus. And normally when you stress an unlesioned animal, you see an increase in FOS in the PVN, where the CRH cells would be, as well as an increase in the expression of CRF mRNA. When you lesion the ventral speculum or ventral hippocampus, you actually see an increase in the amount of FOS and CRF mRNA. This suggests that normally when it's unlesioned, the ventral speculum or ventral hippocampus is actually helping decrease or kind of tune down the stress response in the CRH cells. So what happens when we look more peripherally at the bloodstream? So the CORT release as measured in the blood plasma again shows an increase after you lesion the ventral hippocampus, suggesting that normally the ventral hippocampus helps tamp that down. Interestingly, this seems to be more concentrated in the delayed period during the return to resting state, suggesting maybe this is some kind of off signal that helps turn off the stress response. So that's one of the current models of how the CRH cells are regulated. However, we also know that ventral hippocampus responds to anxiogenic stimuli itself. So for example, in the elevated plus maze, when an animal moves from a closed arm to an open arm, you see an increase in activity of individual cells, as well as an overall increase in activity of the ventral hippocampus. And we know from our own data that at an acute level, measuring with calcium instead of FOS, you can see that the CRH cells are activated independently in a series of foot shocks, similar to the responses of the ventral hippocampus. So maybe there's some kind of GABAergic relay in between these regions, but clearly the model is a little bit more complicated than a simple direct connection. To get at this a little bit further, we decided to see what does a reversible acute inhibition of ventral hippocampus do versus these older studies which use lesions. And we know that lesions have some kinds of off-target effects occasionally. And because they're over many weeks, it can have different effects on the circuit. So when we inhibit using DREADS in the ventral hippocampus, we see, in fact, that in the DREADS condition, there's a lower response in the CRH cells to a stressor versus the control condition shown here. So this runs totally counter to that older model, which is based on FOS activity. And to get at this in another way, we also tried activating the ventral hippocampus. And in this case, we see that activation of the ventral hippocampus using bilateral crimson leads to similar activation of the CRH cells. Again, which all runs counter to that older model based on FOSS. And what does it all mean? Well, we don't know yet, but so far we've kind of come up with the original model, which is that ventral hippocampus inhibits the CRH stress response, and the more acute model, which is that ventral hippocampus contributes excitation to the CRH response, and that's based on calcium activity. So our ongoing experiments are trying to determine whether this is some kind of result of there being separate pathways that lead to either activation or inhibition of subpopulations of CRH cells using single cell imaging. And just to sum up, I want to thank my lab and everyone at UCSF, and I'm happy to take any questions.
Great, thanks, Victoria. So yeah, as a reminder, if you have any questions, feel free to put them in the Q&A. And I guess while we wait for some questions, I guess I'll ask a few first. So it's really interesting. I guess, are there applications in terms of, just not just like measuring the stress response, but also like going in and, I don't know, zapping the hippocampus or some form of that? I know that's probably, the hippocampus does a lot, so you probably don't necessarily want to zap it, but is there some way you can sort of modulate the stress response by modulating hippocampal activity?
Yeah, I think one of the big goals of this research is figuring out how we can kind of apply it to humans eventually. So in terms of mice, of course, we can go in and look for specific cell types. I think that's the most easily translatable part of this research. Maybe if we are profiling the cells and their RNA, then we can find a specific marker that we can use to target a drug. On the other hand, like eventually it may be possible to do things like TMS at a deeper level. The ventral hippocampus is not that close to the surface. So right now, I don't know how feasible that is, but it may be possible to do some kind of therapy. Current therapy for these kinds of like PTSD and trauma-related disorders is really eclectic. And a lot of it is kind of empirically based. So there's this one called eye movement desensitization, which is where you are humans are asked, or patients are asked to kind of recall traumatic memories in a controlled, safe setting and move their eyes in similar, like repeating motions. And this seems to do better than simple like exposure therapy where you recall traumatic memory. And we don't know why that works exactly. Someone, I think a year ago, tried to trace out a circuit in mice that might be related, but yeah, that's kind of like the state of the art in treatments right now, aside from antidepressants. So any kind of clues we can get towards how this is regulated could be really cool.
Interesting. I guess, so you sort of showed like the pathways involved. Does the vagus nerve play any role in that direct pathway, or is that sort of a more high level or different?
Yeah, you're right. That is another therapy that people are trying out right now in terms of trying to reduce stress. I guess, yeah, at some point, I feel like the two axes of stress, the research on them has kind of diverged. So I'm kind of in this boat where we're researching the court-related longer-term effects. Only recently have we found that CRH cells are actually important for like very immediate sub-second behavioral choices. So that has become more complicated. And then on that end with the vagus nerve, you're typically thinking about like controlled breathing, controlled respiration, and heart rate. And those are more thought to be like this other axis, the sympathetic nervous system, which is more adrenaline based. And so I don't know, I guess in a textbook level, we've been taught to think of these as two separate things, but clearly like the neural regions that control both of them are highly interconnected. So it may be that by helping patients or like discovering what controls one axis, we're actually going to be able to control or figure out both axes of stress response at the same time. And yeah, I know that the PBN has reciprocal connections, or these cells have reciprocal connections with, what is it called? Some parts of the brainstem that are really important for controlling heart rate. And I think that's through the vagus nerve.
Oh, yeah, that makes sense. Cool. All right, so we have a question in the Q&A. So it says, great talk. Do you anticipate / observe a rise in court following acute hippocampal activation?
Yeah, that's a great question. We haven't done court measurements, partly because I'm working in mice. And so these older studies, interestingly, of course, they chose to do the court measurements because they were working in rats, and you can do an indwelling catheter. In mice, of course, the advantage is you have these genetic lines, but very little blood. I think they have like 2 ml or something, maybe less of blood. So we hope to look at the court measurements eventually, but we haven't figured out a feasible way to do that yet. Great. And we do anticipate, I guess, that we would see some kind of similar response. It might be that a little bit of activation is not enough, though. So you might have to activate for longer periods of time to see that kind of response.
Yeah, I guess I might have missed it, but like, what's the time scale of the response that we're talking about here?
Yeah.
Some of the stress stuff I'm familiar with is like cortisol, which acts on, probably much longer time scales of like 20 minutes or so.
Mm-hmm. Right. So let's see. I think so. The CRH cells get activated within, milliseconds because the neural circuit is quite quick to activate them, and then they release CRH, which causes the release of ACTH into like the portal vein, and that causes then release of CORT from the adrenal glands. So from beginning to end, I think CORT probably takes a scale of like 5 to 10 minutes, something like that. Quite long for, I don't know. As a neuroscientist, I'm used to studying behaviors that are a little bit shorter. So that's the long time scale we're talking about. And of course, I've been measuring these behaviors and like neural responses that are on the scale of like 2 seconds or something, 3 seconds. So it's interesting to try and figure out how those two differ. Like maybe if you average over many, many seconds, minutes of photometry signal, you actually do see something similar to the CORT responses.
Is that considered a slow timescale, 2 to 3 seconds?
I would say, yeah, relatively. Like I'm used to thinking about just like millisecond, half a second delay between a behavior starting or something else happening. I do know, you're right, now that you say it, there are some regions of the hypothalamus where you read about there being these kind of delay circuits built in where it takes like 5 to 10 seconds for a behavior to appear. And some people have worked out the circuits of how those can function, but it's still, I think it's pretty active area of research, how that happens.
Great. All right, so I think, thanks again, Victoria. I think we're gonna move on to our next speaker. And our second speaker is George Zoidel. He's a master's student in neuroscience at York University in the lab of George R. Zoidel. And his talk is on a role of pannexin 1 channels in an experimental zebrafish model for Parkinson's disease. So whenever you're ready.
Well, thank you for the opportunity to speak here at the NeuroMatch conference. So, why is it not moving? Interesting. Let me try that again. All right. Yeah, there we go. So pannexin 1 is an integral membrane protein, which is important for ATP release. And it's implicated in many different types of diseases, such as epilepsy, stroke, and inflammation through the NLRP3 inflammasome. And the hypothesis in my research has been that the loss of PANX1 function is modulating early stages of Parkinson's disease by affecting the dopaminergic signaling and the inflammasome. So for my Parkinson's disease protocol, I use the acute MTP protocol, which can take effect within a 4-hour incubation period, which models the early stages of Parkinson's disease. So it looks more at the inflammation. What's so great about MTP is that it's able to freely cross the blood-brain barrier, where it's then converted to MPP+, which can be taken up by dopaminergic nerve terminals through dopamine transporters. So right here, I have my real-time PCR data. I've converted it into a heat map. And what we found after MTP treatment is that there was a clear upregulation of Parkinson's disease-related genes, such as PARC7, tyrosine hydroxylase, and VDAC3. We also found that there was a downregulation of genes such as FOSA-B and circadian clock genes like Cry5 and PER3. A second type of experiment we performed was an RNA-seq. And we took the RNA-seq data and put it into gene ontology enrichment analysis, which demonstrated that the inflammasome and energy metabolism were the most significantly upregulated biological processes after the MTP treatment. So we took the regulated genes from the inflammasome and put it into a string, which is a functional protein association network analysis. And it predicts which pathways are regulated through the MTP treatment. So we found right here in green that the NF-kappa B pathway was affected by the MTP treatment. And it's activated by dopamine, which is quite critical for Parkinson's disease. And it primes the NLRP3 inflammasome. Another pathway that was affected was right here in red, the MAP kinase pathway, which regulates the NLRP3 inflammasome. Here in blue, we have more general terms which can be clustered into cell death with the caspase present. A third type of experiment we are doing behavioral genotyping, behavioral testing. And we do this in vivo through pharmacological blocking of the NLRP3 inflammasome. So we're able to block PUNX1A through either a genetic knockout or through the use of probenecid, which is able to block pannexin. And we can also use MCC950 or INF39, which are both able to block the NLRP3 HPase. And this blocks inflammation activation and caspase activation. So right here is the behavioral data for probenecid. And on the left side, you can see in blue the topful longfin, which are the wild type. And on the right in orange, we have the PUNX1A knockout fish. We are measuring the total distance that the fish travel. So we're looking at the locomotion over time periods at different light intensities. So we start off in the dark, and then we slightly ramp up the light every 10 minutes. And as you can see, the movement decreases in the dark. And then as you ramp up the light, the locomotion increases. When we treat the zebrafish with MTP, the movement, the locomotion decreases quite significantly in the dark and in the light for both the TL and the PUNX1A knockouts. When treated with just probenecid right here, you can see that the TL starts to resemble the PUNX1A knockouts. And in a combined treatment of MTP and probenecid, probenecid seems to worsen the MTP-induced locomotor phenotype even further. So some of the highlights from my research so far is that we demonstrated that in an acute MTP model of Parkinson's disease, the loss of function of pannexin-1 channels in the zebrafish significantly upregulated the biological processes of the inflammasome and energy metabolism, demonstrated an upregulation of Parkinson's disease-related genes and a downregulation of circadian clock genes. And we demonstrated that genetic and pharmacological targeting of pannexin-1 worsens the MTP-induced behavioral phenotype, as seen in the probenecid. Also, we demonstrated that the pharmacological targeting of the inflammasome is insufficient to alter the MTP-induced behavioral phenotype. Anyways, thank you for listening to my presentation, and yeah, thank you to everybody in the lab.
Awesome. Thank you, George. would you mind going back to the previous slide? Perfect. So if you have any questions for George, please put them in the Q&A. I have a quick question. So you mentioned that you demonstrated this upregulation of Parkinson's-related genes and downregulation of circadian clock genes.
Yeah.
So I'm not as familiar with Parkinson's disease in terms of genes and all that, but like, is there a relationship between sleep and Parkinson's disease that's well noted?
Yeah, so as I say right here, the circadian arrhythmias are very common in Parkinson's disease patients. So they have problems with sleep.
Mm-hmm.
Yeah.
Interesting. All right, so we have a Q&A question. So the question is, how old were the zebrafish in these experiments? If they were still larvae, you are manipulating things during development. How easily translatable do you think this is to Parkinson's disease, which is usually adult onset?
Yes, that's a good question. So for my experiment specifically, I use 6-day-old zebrafish. So they're still larvae, but they're fully developed. So the brain is fully developed. So there's no difference to the adult zebrafish brain. So it translates more to the early stages of Parkinson's disease. What I'm looking at, since I'm doing the MTP treatment, I'm more looking at the inflammation part, which can later on lead to Parkinson's disease. So I actually have a slide about this later on. Yeah, here. Oh. Okay, there we go. There's a new hypothesis that the Parkinson's actually originates through inflammation in the gut, and then this inflammation travels to the brain. So I'm more focused on the inflammation part. And this inflammation can form Lewy bodies in the brain, and this spreads throughout the brain and causes damage, which is seen later on in Parkinson's disease when dopaminergic neurons are destroyed and there's neurodegeneration. Yeah.
Great. Victoria says that she has a question.
Yeah, I was going to say, I know some people studying how mitochondria might be involved in Parkinson's disease. So it was interesting to see that kind of convergent evidence here. Do you know what, well, first of all, do you think the mitochondria are involved in Parkinson's disease at all? Or like, how does that relate to this Lewy body? And then maybe another separate question is, what does PANX1 do normally?
Okay, that's a good question. So MTP actually inhibits the electron transport chain at the second part. Oh my God, I forgot the name of it. But yeah, it inhibits the second part of the electron transport chain. So it inhibits ATP production. So yes, mitochondria are very critical in this model for Parkinson's disease, since we're stopping the production of ATP. And the normal physiological function of Pannexin-1 is that it's an ATP channel. So under physiological conditions, it is able to release ATP to the outside of the cell, which can then attach to prunergic receptors that can cause prunergic signaling downstream. And then that can cause behavioral and genotypic changes.
Thank you. Great
Thanks.
talk.
Any other questions? So I guess I just have a question about your research in general. Like, is both, you talked about, you know, your interest in exploring the gut-brain axis. Is most of this with this specific ion channel, or sorry, protein channel, or is it with other mechanisms beyond that?
Well, right now I'm looking at Pannixin-1, and it's also expressed in the gut. So it's expressed in a lot of different tissues. So this could play a role in these early stages of the gut-brain axis and the theory that has been put out.
Awesome. Great. All right. Well, I guess we have no other questions. We'll move on to our last speaker then.
I would like to come in and say I can share a splash screen for the next 3 minutes so that the next speaker is on time. So I'll go right ahead and do that.
Thanks.
I think I might be the next speaker because I'm scheduled at 5:30.
Yep. Yeah. So you should start in the next 3 minutes. In the meantime, let's just burn time a little bit, just in case there are more attendees waiting to come at 5:30. That's all. Here we go. I may as well show my face. Oh, I can't show my face. Okay, so I hope everyone's having a good time. Sorry to jump in and all that, but has anyone had a particular favorite thing about the conference or previous conferences, or what you hope for the future for next conference? I can start. Well, I probably really like the keynotes from yesterday. They were, especially the one on brain-machine interface ethics. That was pretty cool. We had some very interesting questions, especially to the IEEE about, neuroscience, how it is involved in the public sector, and those things. I don't know, and I just think that it's really cool that this conference created a space to talk about those kinds of things.
I like to say that it's pretty fun to see how different people are researching different areas of neuroscience that I really haven't been exposed to before. So seeing like all these introductory talks about people's research, it definitely like enlightens the public audience of what's going on in the current specialties within neuroscience.
Mm-hmm. Yeah, it's very cool. There's so much, and like the fact that they were able to fill like these 6 different themes was just astounding. And at that point, I'm going to stop screen sharing, and you should go right on ahead, Vicky.
Yeah, I can introduce you real quick.
Yes.
Yeah, so our final speaker is Vicky Mack, and she is a research assistant at Columbia University in the lab of Aaron L. Barnhart. And her talk is on exploring the role of the endoplasmic reticulum in regulating activity-driven intracellular calcium signaling in Drosophila elementary motion detection. So whenever you're ready.
Sorry, that was a mouthful, I know. Okay. Oh, my bad. Okay, so in the Barnhart lab, our fundamental purpose is to examine the cell biology of neurons. This includes looking at how intracellular activity affects single cell processing and overall circuit function in vivo. If we can understand neuronal processing at the cellular level, imagine the large-scale effects we can see on an animal's brain and behavior if we alter regulatory mechanisms within the neuron. For this project, I'm studying how calcium handling in the endoplasmic reticulum, or the ER, affects neuronal function in the elementary motion detection circuit of the Drosophila. One of the major stores for calcium in the neuron is the ER. The ER stretches throughout the entire neuron, and calcium can flow into and out of the organelle within the cell. Calcium is an important second messenger in neural transmission, since it initiates synaptic vesicle fusion and triggers neurotransmitter release from vesicles. We're focused on understanding how calcium handling in the ER can ultimately shape cytosolic calcium signals and consequently shape neurotransmission, and how ER calcium handling can affect the function of not only that single neuron, but also within an entire circuit. Although the molecular mechanisms driving calcium handling in the ER has been studied in cell cultures and slides preparations, there have been lack of studies showing how this process works in vivo. So, the Drosophila visual system provides a highly stereotyped cellular architecture, since its visual circuit is very well defined. The Drosophila elementary motion detection circuit operates by extracting and comparing luminance signals from adjacent points of space at 2 different points in time. Elementary motion detection is comprised of different types of neurons that detect local motion visual cues. On the left, photoreceptors project onto primary lamina neurons. Lamina neurons split signals into the on and off visual pathways. In the on pathways, secondary medullary neurons depolarize in response to light increments or lights on, and hyperpolarize to light decrements. In the off pathway, these neurons respond in reverse. They depolarize in response to light decrements and hyperpolarize to light increments. Medullary neurons respond differently to visual stimuli, having different response kinetics. In the right figure, the 2 curves represent the responses of 2 different neurons. Notice how one has a faster signal than the other curve. These different timing responses determine whether a signal is either suppressed or enhanced. These third order neurons depend on this temporal filtering to generate direction selective motion signals. These signals are pulled and processed by projection neurons traveling to the central brain. This allows flies to detect different optic flow patterns, such as motion in different directions. We think that calcium handling in the ER contributes to the timing response differences of medullary neurons and affect the temporal filtering of these cells. For this project, my experiments are focused on the MI1 neuron, which belongs to the on pathway and projects onto direction selective T4 neurons. So for my experimental plan, I'm conducting 3 broad experiments on ER-mediated calcium handling in the MI1 neuron. First, we want to know whether there are responses in the ER to visual stimuli in the flies. So first we need to correlate ER and cytosolic calcium signals across different compartments in MI1 neurons. The second experiment is concerning how the ER and cytosolic calcium responses, how they respond at all to molecular perturbations to the ER. And lastly, to determine how the ER contributes to overall circuit function, we want to measure downstream effects within the cytosol of downstream neurons as we perturb the ER in MI1 neurons. So for our experimental setup, the fly's head is dissected first, while the entire fly is melted to plastic holder. The fly is then placed to face the stimulus screen, which will display 2-second light flashes periodically. On the right is a recording of MI1 cytosolic calcium signals to light flash stimuli. As you can see, it just responded very brightly to light flash on, and then it decreases in brightness to light flashes off. Okay, so for our first experimental plan, we want to determine whether the ER does respond to visual stimuli at all. So the MI1 neurons have 3 different compartments. They have dendrites in layers M1 and M5 of the medulla, and they have axons in layers M9 and 10. And so usually in vivo two photon microscopy imaging, we measure cytosolic and ER calcium responses across a period of 4 seconds. As we measure the different fluorescent intensities till light flashes on, indicated in a lighter green or lighter purple color. We also measured responses to light flashing off in darker green, darker purple colors. We could see across the cytosol that calcium responses increase. They have very fast responses, and they also have very sustained responses at the stimulus onset when the light flashes on across all 3 different compartments. So this shows a homogeneity across different compartments. This is a predictable response from what we know about on-pathway visual neurons. But in the ER, this is the first time that we're seeing that the ER does respond to physiological stimuli, but they also respond heterogeneously across all of these compartments. So in layers M1, there is fast transient calcium uptake when the light flashes on. However, in dendrites of layer M5, there is a slow, sustained calcium release in the ER to light flashing on. And in the axon terminals, we see that there is a fast yet sustained calcium uptake at the stimulus onset. So first, we show that the ER does respond to physiological stimuli. Second, these responses are heterogeneous, meaning that this suggests that the ER has different functional needs across different compartments within a single cell type. So for our
yes. Just wanted to butt in real quick. It's been about 8 minutes. I just want to let you know, you might want to wrap up soon.
Oh, sorry. So for the second experiment, we wanted to measure ER and cytosolic calcium responses when we perturb the ER. And so there are different molecular mechanisms that drive ER calcium dynamics. When a signaling ligand attaches itself to a G-coupled protein receptor, this generates IP3. Both IP3 and calcium together activate IP3 receptors located on the ER membrane. These IP3 receptors, when activated, allow for calcium release from the ER into the cytosol. The only way that the ER replenishes its calcium levels is through sarcoplasmic calcium ATPases. So when we knock down, when we knock down IP3R, we wanted to see if this alters ER calcium signals in MI1. So in layers M1, there is not really too large of a discernible differences between both the control and the IP3R RNAi knockdown. In layers M5, however, we can see that calcium uptake and calcium release are both diminished. And in axon terminals for layers M9 and 10, we see that calcium uptake is more sustained in the IP3R RNAi knockdown. And calcium release as well is has been less sustained. So we can see that the IP3R RNAi has subtle effects across all different compartments within a single cell type. But what does that, how does that affect the cytosolic calcium responses? We can see that across all different compartments that cytosolic calcium responses are more sustained when you knock down IP3R receptors. So from these results, we considered that perhaps the ER has a compensatory mechanism in which when there is a deficiency in IP3R activity, then the ER, through some means, is able to decrease circuit activity as well. So there must be a mechanism within the ER that is able to detect calcium levels within the organelle to determine and to modulate both uptake and both release of calcium within the organelle.
I think we're kind of out of time at this point. I think we should probably leave some time for questions. If you could wrap up real quick, that would be great. Just like a quick conclusion.
Yes. After gathering all of these results, we want to later on see how the ER plays a role in mediating calcium signaling downstream in overall circuit function. By understanding how ER plays a role in single cell processing and large scale circuit function, we are able to understand how intracellular activity can have an overall broad scale effects on how we are able to process information from our surroundings and make decisions and think decisions on how we choose to behave to these sensory stimuli. If you have any questions, feel free to let me know, and I'll do my best to answer them. I'd like to thank the Barnhart Lab for supporting me throughout this research project.
Awesome. Fantastic talk. Thanks, Vicky. So we do have a question in the Q&A. It says, nice work. Sorry if I missed this, but how can you differentiate between the cytosolic and endoplasmic reticulum CA2 response? Is your imaging resolution high enough to differentiate those different intracellular compartments?
So it's not really a matter of imaging resolution, actually. It's more of that we use different types of calcium indicators for both the ER, and the cytosol. So let me see. So the ER and the cytosol have different calcium concentration levels. The ER has a cytosolic, the ER has a calcium concentration that is 3 orders of magnitude higher than that of the cytosol. So using that, the Ryan Lab has created a GCAMP calcium indicator for the ER that has a low binding affinity, such that we are able to view 2 different calcium indicators for both the cytosol and the ER in a cell type specific manner.
Great. Awesome. Well, just want to thank all 3 of our speakers again. It's been really interesting talks, some great discussion. Also want to thank Elena for managing the back end of this session. Thanks for everyone for attending. Yeah, I think that's it for this session. Thank you all.
FKBP5 is demethylated by childhood adversity, especially in the group of subjects that have this genotype in this single nucleotide polymorphism. You see that here, like it's a rather half-half, 50-50%. The participants with the CC genotype, they are not so much affected by childhood adversity, but the participants with the risk genotype, their FKBP5 is demethylated by childhood adversity. But that actually is in replication. That was actually shown before. What was not shown before are the neural correlates of this demethylation, what we think is the risk mechanism for psychiatric disorders, especially stress-related psychiatric disorders. And what we found is that FKBP5 DNA methylation is actually pretty strongly correlated to brain structure in this region, in the ventromedial prefrontal cortex. Actually, because FKBP5 is relevant for the cortisol signaling, we actually hypothesized that it could be relevant for the hippocampus or something, but we actually found no correlation there, but a strong correlation here that survived correction for very big mask of a very big region of interest mask. And then we took that brain signal that we got here. We took the sum of all voxels that are correlated with FKBP5 DNA methylation and look whether they are significantly associated with anxiety in our sample. And that was actually the case in those two regions here in the pregenual ACC and in the MOFC medial orbitofrontal cortex, some-ish, I don't know. That was actually significantly associated with trait anxiety in our sample in the way we expected. Okay, so it's negatively associated. So if FKBP5 is demethylated, then the ventromedial prefrontal cortex is, the volume is reduced and trait anxiety is up, heightened. So that is actually why we think that childhood adversity impacts trait anxiety in adulthood. And it's actually partly mediated by the demethylation of FKBP5 and reduced brain volume in the ventromedial prefrontal cortex. That's it. Any questions?
Yeah, I guess this seems to be like, say, uncovering, the goal is to uncover a latent sort of cause of, or I guess the pathway, the mechanistic pathway that may relate the childhood adversity to the trait anxiety. So was this done through like prior knowledge of the pathway, or was this more exploratory that led you to the specific gene into and then the brain area?
It was actually
Does that make sense?
Huh?
Did my question make sense?
It makes total sense, actually. To be honest, it makes so much sense that I prepared 3 extra slides for that question. Because actually it was very hypothesized. Because actually we had epic chips, so we did not have to concentrate on one gene. Could have just like do something like, I don't know, without any hypothesis, but we concentrated on FKBP5 for a reason. And FKBP5, as I said, is relevant for the cortisol signaling because it inhibits the glucocorticoid receptor in the cell. So it dysregulates this glucocorticoid dependent signaling. And a group, as I said, the first part of my talk was a replication, because this group in Munich, they showed this gene environment interaction of this genotype with childhood adversity that converged on the demethylation of FKBP5. And they actually found a molecular mechanism why that is the case. It's the case via cortisol and the chromatin shape. So in the participants with this risk phenotype, or with this risk genotype, the chromatin is shaped in a way that is prone to cortisol that demethylates the gene. And our brain region is actually a region where like these three factors converge. They are particularly relevant, they are particularly altered in mental illness, especially stress-related mental disorders. You see that here the disorder involvement, it's really high here in the medial prefrontal cortex, but also in the hippocampus. And they are prone to stress, again, the medial prefrontal cortex and the hippocampus. And they are involved in the affective regulation, like the medial prefrontal cortex, the perigenual ACC. They are like top-down inhibiting the amygdala and the ventral striatum. Here, mostly serotonergic pyramidal neurons. So that makes sense, because we knew in advance that they are altered by stress, that they are altered in mental disorders. And then if the volume is reduced, they probably can't inhibit, or the inhibition of the amygdala, of the subcortical structures, is insufficient. So it makes sense in this emotion regulatory context. Thank you for the question.
Thanks, Thomas, and thanks, Rona, for the great question. Let's see if we have more questions. So far, we don't have questions coming up yet. This is far away from my research area, but I have a question I can ask. These findings here, can we use them to diagnose or predict the stress-related or some other emotions problem?
You mean like in patients?
Yeah, like PTSD. Can we?
Well, PTSD is actually it's the classic of stress-related mental disorders, and actually this lab that is very famous for FKBP5 for this gene in Munich, they actually proposed something like an FKBP5 depressive subtype, so that like a certain amount of depressive patients actually have like a important dysregulation of the FKBP5 signaling. And they actually developed a specific inhibitor of FKBP5 that was actually there are like a few publications already on this inhibitor that show that it actually has very interesting effects on neuronal tissue cultures, where it induces an enhanced growth of neurids in tissue samples, and that it has anxiolytic properties in mice models of stress, of chronic stress. So again, this anxiety and this neural structural components are actually very interesting and regarding our findings.
Yes, thanks very much. We still have some time.
I have a question.
Cool, sure. Go ahead.
Hey, very nice talk. Looks like an awesome work. I was thinking in this idea of relating genes, specific genes with the spatial mapping of MRI. It was brought to my mind that there's this Allen Brain Atlas. I don't know if you know that it has this microarray to see the expression of different genes. Have you think about relating this specific gene that you found in the expression profile of that atlas with the specific things that you found in the brain structure of gray matter?
Yeah, I'm actually thinking that's actually something that I'm working on right now. I try to Actually, I do have the idea. I thought about that, you know, like extracting the t-values of that map and somehow like relating that to the expression of the genes that are like obviously FKBP5 first, but then the genes that are associated with FKBP5 and just like trying to correlate them with the t-values. I did not do that yet because I actually have to read myself into the pre-processing of the AHBA. And
yeah, there's the NeuroSynth toolbox, which is very useful because it has the gene expression atlas, but there's also different atlases, very useful for doing meta-analysis and these kind of things. So
would be interesting. Yeah, could you send me something about it?
Yeah, of course.
It'd be awesome. Thank you very much.
Okay.
Cool.
Do you have questions?
Do I have to stop share?
Yes, I think we're almost done. Almost the time for the next speaker. Thanks very much again, Thomas, for your wonderful talk and discussion. Our next speaker is Ronan Perry. Ronan obtained his master's degree in biomedical engineering from Johns Hopkins University with Dr. Joshua Wagerston. He's currently a research assistant in the same group. He's also a lead developer of the Python package MVLearn. Please do check more information of him in the chat. Take the stage, Rona, please.
Okay, you can hear me all right?
Yes.
Yes, thank you for the introduction. And I'm going to talk a little bit about my work on permutation corrected independence testing with respect to high dimensional fMRI data. So first of all, I just want to give acknowledgements to all of my collaborators on this project. So our collaborators at the CRNL for their work on obtaining the data that I'll talk about, as well as Daniel Margulies and Joshua Vogelstein for their role in the methodology. So this project really began out of the motivation from the data that we had on hand. So the data consisted of 86 subjects, all expert or novice meditators. So expert meditators had over 10,000 hours of experience, whereas novice meditators had just a weekend of training. And each of them were measured over the course of 10 minutes in 3 different states, either resting or the open monitoring meditative state or the compassion meditative state. And this amounted to a total of 228 scans over all these subjects. And the resolution is quite high. So it's at the cortical vertex level. So it's over about 18,000 cortical vertices. So it's pretty high dimensional in that respect. And this leads to some natural questions that our collaborators were interested in. Say, are there differences we can see across, say, expertise? Are novices and experts different? Or maybe novices, are they different in resting versus meditating? And so the first sort of part of this project and the work is really extracting a low dimensional representation. So there's a term called cortical gradients, and cortical gradients are these low dimensional spatial maps of the brain. So every voxel takes on a univariate value, and things that are close to one another in the spatial map mimic each other in the temporal processes. So you really reality to capture sort of shared temporal processes. It's similar. You can take linear approaches that'll mimic, say, PCA, or you can take very nonlinear approaches that get a little bit more complicated. And so through this dimensionality process, we reduce each subject in each state from this large multivariate representation to a single gradient, or actually we compute 3 gradients for each subject, which encapsulates some of the functioning. And so here I just show, an example of when you embed it into this low dimensional representation across all the different expertises the average. So you get these sort of gradients that show which parts of the measured brain are, behaving similarly temporally. And the dimensionality reduction was performed using a generalized canonical correlation methodology. And so that's going to find patterns that maximize correlation over all 228 scans. And I just want to give a mention to toolkit MVLearn, which is where this the tool came from. I'm one of the lead developers on it, and it's really just a collection of Python tools for multi-view data. So if you have, say in Thomas's case, you might have data from a brain and from genotyping, and you want to find patterns that maximize their correlation. And so we have a bunch of different tools, but I just wanted to mention that. But continuing on, so after we have these sort of low-dimensional representations of the data, it's multivariate still. We want to go back to original question. Are there, say, differences between experts and novices? And we can frame this in terms of independence test or a quality of distribution test. So maybe we have the distribution of the gradients, these low-dimensional representations of the experts, the novices, and now we want to say, are they equivalent or are they not equivalent? And in this case, a t-test would look at individual voxels. We're trying to look at the entire gradient, so all 18,000 voxels at once. And so we use this non-parametric multivariate test called distance correlation. And that basically just looks at the distance matrix, and it uses sort of a weighted sum of the distance matrix to say, is this big, is it small? And that'll tell you after doing a permutation test, whether that's, say, abnormal for these labels or not. And sort of part of my talk is just about correcting for exchangeability. So if you remember, in this case, we actually had measurements for the same subject. And so naively, one might try to apply a test assuming IID, so independent and identically distributed, but actually, because we have repeat measurements from the same subject, you can't assume that independence. So when you run this test here, you have to keep that in mind. There's actually this hierarchy of dependence in your data where you have the same subjects and then each of them have different measurements. And so if you're familiar with permutation tests, basically you just shuffle the labels of your data and you calculate the same test statistic. You can't just do that with any permutation because some aren't valid ones that you might see in your actual data. So this is sort of a crucial aspect. If you don't correct for exchangeability, your p-values get messed up. But correcting for this, we are able to run this distance correlation first at sort of a 6 sample. So if you're familiar with like ANOVA tests, you can start with a multi-sample sort of test and then work your way down, going from F tests to T tests. So here is similar. We first ask, are there any differences between all of our states and expertise levels? And so in this top row, we see significant p-values at certain gradients highlighted in X. And we control for the multiple testing using the Bonferroni correction, which is the most conservative that you can get. And we see some significances primarily in gradient 3. And then we can look down into the 3 sample, just our experts in their states, and then look at all these pairwise comparisons. So we can ask, in this row, say, are all the individuals resting different from all of them in the compassion meditative stance? And that seems to be the primary source of differences within our low dimensional representation. And that would lead to future questions of ours. And that's sort of where we are currently with the work. So I just want to say thank you for listening and just any points of discussion, whether the cortical gradients, distance correlation, hypothesis testing in general, or MVLearn. But I'm free to take any of your questions now. Thank you.
Thanks very much, Rona, for the great talk. Currently, we don't have questions coming up. Thomas and Vincent, do you have questions for our Ronan?
Yes, I have a question. Ronan, great talk. I was wondering, as you mentioned in at the first slides of your talk, that the cortical gradients could be observed also with other methods. Why did you specifically use this method? Because I've seen like most commonly the PCA method. And
Yes.
as another question also, does these cortical gradients spatially correspond with, I don't know, maybe canonical networks or this kind of things that has also been used with the cortical gradients literature?
Yeah, so as to the first question, I guess, yeah, there's a bunch of different ways to compute it. So the way we were doing was, there's a couple different names for it. It's actually, you can call it like group PCA, or it's a form of sort of reduced canonical correlation analysis, but it is actually based on sort of linear methods like PCA. If you just applied so we call it a multi-view method, because it's, rather than say applying PCA to each measurement independently, you have to, if you just did that, PCA is sort of invariant. There's some sign and rotations that there's some invariances going on there, and you wouldn't really, what you would find, they wouldn't be aligned correctly. And so this sort of not only does that dimensionality reduction, but makes sure they're all aligned into the same space to maximize that correlation. A lot of people tend to use, but other people tend to use, say, diffusion mapping, which is this like fancy non-linear sort of method. But you have to always sort of align your dimension reduction. And yes, and then your second point, people have definitely looked into sort of what the spatial maps of these cortical gradients reveal. For our work, we haven't really looked at sort of what they span, what, say, in red at one end of the spectrum, what that might highlight versus the other end. But there's been lots of prior work on like the HCP data set looking at cortical gradients and showing that they tend to span, say, sensory sensory regions to, say, motor regions, but we haven't looked into that in our work yet.
Thanks very much. Thanks, Vincent, for your great questions. We still have some time.
I will ask a question then. Like, actually, very impressive talk. I'm very impressed. Was huge fun. Well, actually, I don't have like a specific question, but I would actually like to hear more about your MVLearn package, because you actually hinted into some usability for me, obviously.
Yeah.
So,
yeah. So I guess if you're familiar with one of the so MVLearn was simply came about because of this project, but you may be familiar with, there's a set of methods called canonical correlation methods, where you look for, say, if you're familiar with PCA just looks for reduction that maximizes the variance among the components. Canonical correlation may ask, if you have two sets of data, you want a reduction of both that maximizes the correlation. So maybe you have, say, a question in psychology might be, if you have like two different sets of like quiz scores, and you want to find the, say, reduction that maximizes their correlation. So maybe if you have, I don't know, example of like, if you have like trauma scores and say behavioral risk scores, and you want to see which ones sort of overlap the most, you might use canonical correlation to find loadings that sort of give you that. But there was a paper I read about using canonical correlation to, say, look at genotypic data, and then you also had neural imaging data, and you want to find regions that maximize their correlation between the two. And I've also seen this in context of sort of psychological, I guess,
I've seen definitely
quizzes and stuff. But so, for example, you may look at, if you have, so this is just the website's at mvlearn.github.io, but if you have, say, I don't know, maybe the plot's a little confusing actually. But if you have your complex data and it sort of learns projections of it that are maximally correlated with one another. I don't have a great one here that's sort of applied to, say, psychology scores or anything, but the literature on canonical correlation analysis with those applications is pretty broad in my experience.
I'll definitely look into it. Thank you, actually.
Okay, very cool. Very great work. And how about we just move on? Our next speaker is Vincent Madel. Vincent is a PhD student, is a PhD candidate currently from Pontificia University Católica de Chile. In 2017, he joined the Neurodynamics of Cognition lab in San Diego, Chile. His research area is attention and consciousness with a focus on the dynamics of information processing in the brain from a multi-skill perspective, trying to generate the bridges between cognitive and computational neuroscience. That's wonderful. Please go ahead, Vincent.
Okay. Thank you very much. Can you hear me?
Yes.
Okay.
Very well.
And the presentation looks fine. Okay. So I'm going to talk about a bioRxiv preprint that we have with our lab. This is complexity and the 1/f slope jointly reflect cortical states across different excitatory and inhibitory balances. This is a work done in collaboration with Martin Irani, which is the first co-author, and Tommaso Sandone and Gonzalo Bonconte, which are the PI and postdocs in our labs. So as the main idea of this work, we are analyzing, when you're analyzing electroencephalogram or local field potential neural signals, there are different characteristic or specific properties in those time series, in those signals. On the one hand, there's complexity, which calculates the number of diversity of neural time series by binarizing the continuous time to discrete events of brain activity. And here, for example, we have a neural time series, and we just draw the median of all the values in the time series. And if the signal is in the upper side of the median, there's a 1, and if it is in the lower side of the median, it's a 0. So then you have a binarized signal. And the Lempel-Zip complexity represents the repertoire or the different patterns of this binarized signal. So for example, if you have only 0, 1, 0, 1, 0, 1 in all the time series, you have a really low complexity because all your signal could be reduced to only the 0 and the 1, and then you can reproduce all your signal. And the repertoire of the signal, the complexity of the signal has been shown to be related with unconsciousness, with sleep, and also with anesthesia. This is in the time domain, but if you transform the time series into the frequency domain with canonical Fourier transform, there are other components of the signal. For example, if you are transforming your signal, which is a flat white noise, for example, and you calculate the Fourier transform, you can see that in the log space, this looks like a flat spectrum. However, in a more physiological range, if you transform your time series to a frequency domain, you can see a 1/f decay, which represents a very physiological signal, and it's very common in different natural phenomena. And also the brain activity displays a dominant 1/f like signal. And this 1/f slope or decay has been shown to reflect excitatory inhibitory balance, has been shown also to be related with sleep, with anesthesia. But here we ask, how does the 1/f slope and the Lempel-Zip complexity relate in the brain? How does this time series property complexity, right, is related with these frequency specific properties like the 1/f slope? And to do this, we generate, first of all, synthetic signals. We simulate a power spectral density Here we have amplitude or power, and here in the x-axis we have frequency, and we simulated different power spectral densities with different slopes. And then we do an inverse Fourier, which transforms this frequency domain to the temporal domain, and we calculate the complexity, the Lempel-Sieve complexity. And now that we have both the Lempel-Sieve complexity and the 1/f slope of each simulated signal, we can calculate how those two measures relate. And we fit these two measures. And the best fitting equation was a decreasing monotonical function that it was showing that this relation is not linear, but it's actually non-linear relation, which was kind of was very interesting for us. And then we wanted to see how this relates with more physiological signals. So we simulated LFP, local field potential signals, with as independent components of excitation and inhibition, which here's an assumption of the model. If you sum both of these signals linearly, you can get the LFP. This is a conductance-based simulation. And in the same way with the temporal signal, we calculated also the Lempel-Sieve complexity in this more neural-like signal. And we also calculated the 1/f slope of the spectrum. This was done with the FUF toolbox. And we found that actually the Lempel-Sieve complexity and the 1/f slope were both very intimately related, and they were also best explained by this monotonically decreasing equation. And they were tracked, the different state of this relation was tracked by the different states of the excitatory inhibitory balance, because we generated different signals with different levels of inhibition, of the conductance of inhibition. So actually manipulating the excitatory inhibitory balance towards inhibition was changing both the Lempel-Sieve complexity and the 1/f slope. And then finally, to test the model's prediction, right, that this both of these signals of these signal properties have this strange shape, like this decreasing monotonical shape. We went to see a human electroencephalogram and monkey ECOG. This is open data. And this data was in the resting state condition and with propofol sedation condition. The propofol is an GABAergic agonist. So we were assuming that actually with propofol sedation, we were shifting the excitatory inhibitory balance towards inhibition. And we see that each of these points is an electrode. All the values in the 1/f slope and also in the Lempel-Sieve complexity were highly changed in anesthesia, right, in propofol sedation. And if we plot both in human and in monkey signals, and if we visualize how they distribute across all the excitatory inhibitory states, awake in anesthesia, in both human and monkey, they represent also this very nice equation. So as a conclusion and future directions, in synthetic signals, we showed that the relation between the power law exponent, or the 1/f slope, right, and complexity is nonlinear. And we showed that modeling the equation is not affected by the beginning or the end of the power law. This is not shown in the presentation, but if you want to see more about this, you can check the paper in bioRxiv. In the other model, in the LFP model, we showed that altering the balance between excitation inhibition does not modulate the 1/f offset. This is also not shown in the presentation, but it's shown in the bioRxiv specifically, but it strongly modulates the 1/f slope and the complexity. And this nonlinear relation is tracked by the varying states of excitatory inhibitory ratios. And finally, we validate our modeling predictions in macaque electrocorticogram, which is invasive electrophysiology, and also in human electroencephalogram, which is non-invasive electrophysiology, with pharmacological manipulations of excitation and inhibition. thank you very much to Tommaso Sandon, to Martin Irani and Gonzalo Bonconte, co-authors of this work. And if you want to see more about our work, you can check our lab page and follow me on Twitter. Thank you very much.
Thanks very much, Vincent, for your excellent talk. It's good to see the converging evidence with different methods. Any questions for Vincent?
Yes, I would like to ask a question. It was really good. Thank you, Vincent. So my first question is that why this measure of complexity? Because there are many different measures of complexity, like multi-scale entropy. And so why this one? Why the LZ?
Yeah, that's actually a good question, because there are different there are different complexity algorithms like Kolmogorov complexity, entropy. Specifically, this is not like I didn't talk about this in the presentation, but in our lab we have been exploring a lot of the complexity algorithms and entropy algorithms, and we found that specifically Lempel-Sieve complexity is not very sensitive to oscillations. So actually, we found that other types of algorithms like sample entropy, for example, is more sensitive to, for example, an oscillation in the spectrum. For example, we can think about a massive alpha band power in the spectrum. And we wanted to see maybe this Lempel-Sieve complexity is very sensitive to the slope, right? And that was what we were exploring here. Also, Lempel-Sieve complexity is really, low cost computationally. And in the literature, there are very interesting work relating Lempel-Sieve complexity with the transition entropy, like the repertoire of the neural signal. So I don't know if that answers your question, but thank you very much for the question.
Thank you. It's not the last one. I have another question. So I know that people with Alzheimer's, so if you measure their complexity, but using the multi-scale or estimated entropy, and then so people with pathological brain waves, like for the EEG or ECOG, then you see that their complexity does not match healthy humans. Have you also considered this, because the anesthesia kind of points to the same direction, but have you considered studying pathological data to see if you can see something?
Yes, actually, we are considering it, and also there's a very nice work recently by the group of Bradley Wojtek, who works with the 1ORF slope and characterizing this 1ORF in different signals, and he found that it was a really good biomarker for Parkinson's disease. So if the work that we have been studying here, which is the relation between the 1ORF and Lempel-Sieve complexity, is true in all the signals, right? Then we would expect that actually in those patients, the Lempel-Sieve complexity will also be very informative to distinguish healthy subjects from the case study. And also that the very interesting thing for us is that we want to give a higher interpretability to complexity algorithms, because at least in biomedical signaling engineering and these kind of things, a lot of complexity algorithms are being used to distinguish, for example, conscious or to non-conscious in anesthetic surgery and these kind of things. However, there's not a common understanding on what are the underlying physiological mechanisms that are making the signal to change and to be more complex or low complex. So that's what we are interested in, to bridge a gap between more like sophisticated signals like the 1ORF, right? That is frequency domain, and you have to transform the Fourier, etc. With this like very simple algorithms like Lempel-Sieve complexity that is very low cost and is very fast. Also, could be very useful in medicine, for example.
Okay, thank you. Because now at least I know that there is also, I didn't know that we can use complexity for diagnosing Parkinson. That's really good to know. And I work on causality, and we use also Fourier transfer, and like 1/f, it's all over the place. So like that you mentioned, that it's good to know also the direction of, like, we call it the flow of causality. So that's really nice to see that this topic comes up again. Thank you.
That's great.
It was really good. Thank you.
Thank you very much.
Thanks very much for your question, Sid. I think we did a good job to stick on time. Thanks very much again to all our speakers and all the great questions. I think we almost done. Thanks very much, everyone. Thomas, sorry, I forgot to introduce you in the beginning.
No problem. No problem. Didn't even notice.
Thank you, everyone.
Thank you. It was huge fun, actually. Very impressive talks.
Yes, exactly.
Yeah, thank you, everyone, and good to meet you online. Nice to meet you online.
Yes.
Thank you too. Have a nice evening or have a good morning.
Yeah, enjoy the rest of the conference.
Thank you
Thank you
for
very much.
hosting.
Yeah,
Bye-bye.
thank you for hosting. Ciao.
Bye. Thank you.